I think we have all got to know each other yesterday, so I don't need to introduce myself, but I would like to still give a type of introduction from the perspective of Kat as one of the three co-organizers of this workshop in Trust Your Gut. I think from the big perspective, we are really talking about the revolutionary changes which are taking place at the moment in cell culture. We simply have to admit that access to primary cells is very limited, quality and quantity-wise, so human cell systems are difficult to obtain, or have been difficult. And the tumor cell lines which we used as a surrogate were not doing the trick, really. The game changer has been really the advent of stem cell technologies, which allow us increasingly to reproduce human tissues and organs, and the bioengineering of them to microphysiological systems. And I don't think I need to tell you, they come in different flavors, and we have seen some examples for the gut yesterday. And we're also trying to more and more bring them together in multi-organ systems with perfusion. I think this is really a very important step because it is reversing this trend of coming to reductionistic systems, which are simpler and simpler, of cell systems, which have advantages of throughput, but which are not reflecting anymore the complexity of an organism. We're trying to reverse this to some, or are able to reverse this now with more and more organ interactions, organ functionalities, but we are obviously not yet complete. Our center has been following this. Why is it automatically moving forward? Has been following this course. We did these two workshops, for example, where we brought together 35 and 45 experts from all over the world to follow what these technologies are doing. The second of these workshops, actually this was a workshop in the same series as the one we are organizing here. These are workshops carried out under the Transatlantic Think Tank for Toxicology. The second of these workshops led to a science article, which is giving the opportunity to read just 10% and the key information of the workshop. It is entitled Human Microphysiological Systems for Drug Development, and this is where we at the moment see the biggest opportunities for for microphysiological systems. And as a side effect of this, we discussed at the second workshop between Dan Tagle and myself, what is happening here? Okay. Okay. So not in the, okay, we go back. Okay. So Dan Tagle and myself in 2019 discussed that we actually would need to organize this movement further. And we came up with the idea of creating world conferences, world summits actually in the end. And we carried out the first of these world summits on microphysiological systems in New Orleans. And you see Susie Fitzpatrick as one of the three co-hosts, familiar faces here, can be fast. Already then we had about 52 organizations supporting this, 430 people online, 430 people in person and 215 online. 65 participants came from the FDA, showing you the enormous interest in this. But the second one of these, carried out in June of this year, very much increased this. We are now at 60 organizations. We had 96 sponsors for this event. And you see that we actually had to stop at 1,300 participants because the capacity of the venue did not allow us to go bigger. So we stopped advertising it 4 months before the event, showing you the enormous interest. We had a waiting list of people, even a waiting list for sponsors, which I have never seen in a conference. This conference is now going to Seattle. So next June, we will have an event here, and you see with Dan Tagle, we have also a very prominent promoter of these things as one of the co-hosts. So he is one of the three. We are just opening up for the abstract submissions, so there's an opportunity to work here. For me, a lot of this is really into developing, first of all, the physiology, reflecting actual human physiology, but then also pathophysiology. We see increasingly that these models allow toxicity study, infection studies, and other ways of creating diseases. Genetic backgrounds can be introduced by patient MPS and molecular biology. So it is really lending itself primarily to drug development. For toxicology, there's a certain use already now for the non-regulatory areas. This is another T4 workshop we carried out on investigative drug toxicology, and this was addressing how these type of systems and other technologies can be used to early identify, to front load toxicity assessments, to early identify are there problems associated with the target, can we already identify possible liabilities of these substance classes or of individual substances before they even go into the regulatory testing. So this is a tremendous opportunity which is largely used by the pharmaceutical companies in the world, which is the majority of the market for microphysiological systems. There's a second piece. A lot of toxicology work in pharma is de-risking, which means things which have been found in animal studies, which need to be addressed before or while going into these clinical trials. So a lot of effort is spent to show that it is not relevant what these animal models have been showing. And this de-risking very often makes use of microphysiological systems. And what is really liked by both the regulatory side and the companies is also to create the microphysiological systems of the animals to compare, to show that the effect is present in the microphysiological system, let's say of the rat, where the observation was done, but then not found in the corresponding human system. So there's a lot of opportunity here in this field, and I think this is the reason, or part of the reason, why in vitro toxicology is already 3 times as much of turnover than in vivo toxicology. We all perceive toxicology as very much animal-driven because the regulatory side is like this, but the industry is really investing a lot into others. So why are we here? It's in essence because Susie Fitzpatrick asked me several times, what is in the box for regulators? We discussed several times that it is necessary to really ask after these wonderful conferences, what did we learn? What can we actually use for the regulatory process? And the standard answer is always, yeah, everything which is mechanistic, you cannot study really in animals, but you can do it in these systems, and this adds weight of evidence. But this workshop is asking more concrete. For one area, the gut, which we have chosen as an example, which was also Susie's proposal, we developed this workshop to ask, is it possible really also in the regulatory field to make any use of these? And 3 topics were identified, which we discussed yesterday. So I'm bringing coal to charcoal to Newcastle. First of all, is it possible by kinetic aspects of these models to possibly de-risk systemic toxicity? This is certainly not something for drug development, but in the areas where FDA and others are having no pre-marketing authority, but are evaluating models. Is it possible here, for example, to say something which is not taken up in the gut is not toxic? On a systemic level, we can spare some of these study efforts. The second point is also, I think, quite clear. Is it possible to predict GI side effects with these models, so direct gut toxicities, which is certainly very interesting, especially as these animal models are notorious for not identifying them, and many of them lead to problems in the clinical phase of drug development, sometimes to the need to abandon certain entities because of the non-tolerance of these side effects. It would be very interesting in a field where there's essentially no animal model available to understand whether these models are helping. And the third and last point we are focusing on here is allergenicity. While we have some animal models for skin sensitization, we already don't have agreed models for respiratory sensitization, and the food allergenicity is very, very poor. And it would be very interesting to see whether models could be developed for this aspect of work. So I think we are, on the one hand here, witnessing a revolution in scientific methodologies, but on the other side, I think we also from yesterday's discussion had a bit of a sobering effect, because as soon as we were starting to discuss in any detail, how can this really be used in a regulatory context, there is possibilities, but there's also a lot of hurdles. And this is what we are trying to address within this workshop. So these were, let's say, my 2 cents from a, let's say, introductory perspective. And I hope that today's discussions following the talks we have on the list, and then a review of what was discussed yesterday and a path forward towards the report and possibly other activities is giving us a fruitful outcome of this workshop. Thank you very much. So, and with this, I think we can move on. Any questions? I don't think it was not really meant for this, but I'm happy to entertain if there's a quick question or so. Good, the better, because it brings us closer to time again. So we have then on our agenda the first talks. I did the mistake not bringing my agenda. Did you have it at 10? Okay, good. Good, excellent. Good, so the first talk is by John Wambo. We have got to know each other already yesterday, so I don't think it can be very quick. He is, as you will know, an exposure specialist at EPA, and it's your floor.
Thank you. All right, good morning, everyone. Thank you very much for the invitation to speak. I'll be talking today about how mathematically we have incorporated in vitro gut data into some risk prioritization schemes. I do need to say up front, you know, I am a scientist at the EPA. I'm speaking to you as a scientist at the EPA, but I'm not speaking for the EPA. I don't have that power. I don't want that power. So everything I tell you is what I told you, not what EPA told you. So, and I have no conflicts of interest to declare. If you'll excuse me for the first third of the talk, I'm going to describe the scheme that we've integrated the gut data into. Then I will describe how we integrated the gut data themselves and how we integrated it. And so the basic idea is that we have what a 40-year-old guidance from the National Academies of Science, Engineering, and Mathematics that when we consider public health risk from a chemical, we need to consider not just the hazard, but also the exposure and really the dose-response relationship. How do the exposures relate to what we receive? And I'm going to stop getting that microphone, I promise. But that is how we, that's why we're interested in the gut. That's a route of exposure. As was just said, if we don't absorb it, maybe we don't need to worry about the hazard. And so all three of these components are needed to consider risk. And we're interested in next generation risk assessment. What can we do using higher throughput tools to address a wide legacy of chemicals at the EPA that have never had in vivo data generated or have limited in vivo data, and novel chemicals. The EPA gets 2,000 applications for new uses of a chemical every year. How do we test those? And so next generation risk assessment is built upon NAMs. I don't think that's a controversial statement, but at Tox 21, over the last decade and a half, they've screened 8,000 chemicals across about 50 endpoints. At EPA, another 4,000 chemicals have been screened at greater than 2,000 assay endpoints. So that's a lot of in vitro data, but the form that data often takes, it is concentration response, but it's something like, we have seen estrogenicity for this compound at 2 micromolar. Well, what do you do with that? You know, that's interesting, and I'm not denying that that's interesting, but back to that idea that risk is hazard, exposure, and dose response. How do we get the exposure and the dose response for that? And that's really in vitro and vivo extrapolation. And again, I apologize. I know everybody in the room could probably give these slides, but I just wanted to give a baseline. And so in vitro and vivo extrapolations, when we want to take in vitro data and extrapolate it to predict something in vivo. And so that consists of considering in vitro disposition. You know, how does that 3 micromolar nominal concentration differ from the actual concentration in the cells? It includes pharmacokinetics, toxicokinetics, where does the chemical go in the body, how does it leave, and also pharmacodynamics and toxicodynamics. So what does it do to the body, how does it perturb the function of the basic systems of the body? Real issue that I'm going to talk about today is that most chemicals lack toxicokinetic data. So even if you believe me that we need to know toxicokinetics to link exposure and hazard, as we have gone through the ToxGas program, we have found that about 5% of the chemicals tested with the in vitro hazard NAMs have in vivo toxicokinetic data available. So that means 95% of the time we don't have the numbers that we need to put it in a public health risk context. And so my colleagues, Barbara Wetmore and Rusty Thomas, first at the Hamner, now at the EPA, have spent the last 15 years developing tools to basically adapt techniques from the pharmaceutical industry that were designed to evaluate the, prospectively evaluate the effectiveness of a clinical study, design those tools to come up with ballpark numbers for environmental chemicals that have no other toxicokinetic data. And what we call this high throughput toxicokinetics. It's not very clever, but that's what we went for. And the idea is to generate toxicokinetic relevant information for large numbers of chemicals. And these methods, like I said, have been used to design clinical trials. They're thought for a pharma-like compound to typically get you within about a factor of 3 of what you'd actually see in vivo. They've not been that accurate for environmental chemicals, but they're close. And so our primary goal is to give this in a human dose context. And then we have a secondary goal of making everything open access, audible, because we are making risk regulatory decisions. Hopefully using these tools, we want people to be able to review the data, how we got there. And so traditional in vitro and vivo extrapolation, as I think most of us think about it, if you had a drug lead and you knew this is a high impact drug, this is going to make life-altering health improvements for people, or the billion-dollar drug lead, or whatever it was, you hammer it with everything you have to figure out how do I go from in vitro to in vivo. And that often involves using in vivo toxicokinetic data. However, like I just said, so you go from the in vitro concentrations, you work back to internal tissue concentrations, and then you consider what size of pill, or do I need an inhaler, or do I need to do intravenous injection? And that would be based on in vivo toxicokinetic data and our understanding of toxicokinetics. And so what we're talking about with high throughput toxicokinetics is a second type of in vitro and vivo extrapolation, where we're using chemical specific data, but they're measured in vitro to generate our toxicokinetics. And then we're using that toxicokinetics to go from the in vitro hazard point of departure back to still an in vivo exposure or an in vivo tissue concentration. So real short, in vitro or high throughput toxicokinetics consists of in vitro measurements that we can do rapidly. It has to be something that we can measure for hundreds to thousands of chemicals, because Tox21 has measured thousands of chemicals. We need to be able to extrapolate for them. So it's in vitro assays that we can run quickly, and there's been a series of publications describing these data, plus generic toxicokinetic models that I can plug those data into to make the sort of predictions I need. I need to be able to use the in vitro data that were generated. And we've generated a series of what we call high throughput pharmacokinetic models that can take that data and make a prediction. And so those two concepts together, things that we can measure for hundreds to thousands of chemicals, and then models that can make use of that to make predictions, that's HTTK. And this is complicated. We've worked on this for 15 years. I've worked with dozens of very bright people on it. However, what I would like to assure you is at the end of the day, our goal is to do a very simple conversion. And so what we want to calculate is what we are currently calling the administered equivalent dose. Basically, what dose do you have to give someone in order to cause that 3 micromolar that I saw in vitro? And all of HTTK, if we're doing it right, boils down to a single number. It's this factor for in vitro and vivo extrapolation. I have this in vitro concentration, 3 micromolar, and I multiply it by that factor. And I just want to know what that factor is. And so, yes, there's lots of work to generate that factor, and we're continuing to revise it. But that, at the end of the day, that's our in vitro and vivo extrapolation by scaling factor. And that factor turns out to be, or at least as we use it, the steady state concentration for the highest 95th percentile who are receiving the same dose get a really high blood level in the US population. And so it's 1 over that actually, but basically we use that, we calculate that number with HTTK, the steady state concentration for a 1 mg per kg day exposure. You can take your 3 micromolar, divide it by that steady state concentration, and you now have a dose. How many mgs per kg per day does the average person need, or actually not the average, the 95th percentile most sensitive person need to get 3 micromolar in their blood? And so this allows us to do rapid chemical prioritization. So what we have here are results looking at chemicals monitored by the Centers for Disease Control in the blood of average Americans. So all of these chemicals have been, not all of them, but several of them have been tested through ToxGas. So we have at the top in vitro concentrations that have been converted to mg per kg per day. And you see you get a range of activity because, like I said, we've run over 2,000 assay endpoints. So you get a range of points of departure. But if you look at the doses, as you do the in vitro and vivo extrapolation, you have doses here where we're talking up like 100 milligrams per kilogram per day. So I mean, I'm eating a candy bar solidly made out of that. Well, no, that would be like a big salad bowl of nothing but that chemical every day to cause my blood levels. And then down here, we're down at micrograms per kilogram per day, which is a reasonable environmental exposure. When we compare that with the exposure rates that we infer from the CDC, that's what's in orange here, there's uncertainty again. You can see that there's a bunch of chemicals over here where we literally, and this is a log 10 scale, have 100 million times difference between the in vitro dose, the in vitro estimated dose, and the exposure dose. So if I were a graduate student, I'd feel really sad if I were studying the chemical over here because yes, it causes something in vitro, but the dose is not occurring in the population. Now, occupationally, if it's my job to clean the bin that they make this chemical in a factory, I might get that exposure. So this is general population, but still that's less interesting. Then we have a couple chemicals over here where, given the crude techniques we've used, so there's definitely room for refinement, but we have a couple of chemicals where it's plausible that some portion of the population's on average receiving a dose that causes the bioactivity observed in ToxGas. Now, what is that bioactivity? That literally could just be induction of a CYP. That's not adverse. It's not adversity, but it's bioactivity. So anyhow, these are more interesting chemicals on the left than they are on the right. So this is the scheme that we are introducing in vitro gut absorption data into. So my colleague, Dr. Elena Kenyon, presented yesterday on an EPA activity to generate Keiko2 membrane permeability data for non-pharmaceutical chemicals. And so, HTTK really is limited by the processes that we can characterize in vitro, but we now have a lot of data. I mean, this is data on about 3,000 chemicals. And so you can see in this bluish color here, so it's the permeabilities that were reported by Dr. Kenyon yesterday from EPA and the non-pharmaceuticals. And then we have a bunch of data from a couple of papers, and then Chembol is an online repository. Almost all of that is on pharmaceuticals. So, it's still heavily skewed toward pharmaceuticals, but we have a lot of data now on particular chemicals. And so we have a lot of data, but we don't have data on every chemical that the EPA is concerned with. So one of the first things we did is we used those data to build a quantitative structure property relationship model. We used the method of random forest. It's a machine learning model, and tried to train the model not to perfectly predict Keiko2, but just to predict the bin that things were in. And so we looked at things from, can you just predict high versus low? So that's a 2-bin model. We tried to do a 5-bin model of, okay, really high, medium, really low. And what we saw as you look at the accuracy, we were pretty good at the high-low model, but it wasn't terribly distinguishing between the chemicals. We were lousy at a 5-bin model. And so we ended up actually settling on a 3-bin model where it's high, medium, or low. They're nicely separated at 0.2, 2, and 20. And that's centimeters times 10 per second times 10 to the negative 6. So we have a model that can predict these. It's okay accurate. It's not great. And it gives us an ability for chemicals where we don't have measured Keiko2 to start to try to predict what the permeability is. Is it low, medium, or high? And this is just an evaluation set. And you see there's overlap. It's not perfectly distinction. It only predicts 3 values here. And most of the ones in the high are high, most of the ones in the low are low, but there's definitely some, it's 68% balanced accuracy. We then inserted this into HTTK. HTTK already included first pass metabolism in most of our models. So that's where the gut absorbs the chemical from the body, but then it first goes through the liver. The liver is kind of our secret serviceman. Its job is to jump in front of the bullet, so the blood goes through, and we have in vitro metabolism data. So this fraction that's hepatically metabolized through first pass, we already had that in the model. But now with Keiko2, we can predict the fraction absorbed from the gut and the fraction escaping kind of first pass metabolism in the gut metabolism. And we use just literature models for that. So previously we'd assumed that these 2 first fractions were 1. We add that into HTTK, and now we start to look at how well it works. And fortunately, there is a lot of literature in vivo data to compare to, or at least some. So if we look at our ability to predict first pass hepatic metabolism, And here we have pharmaceuticals as triangles and non-pharmaceuticals in circles. We actually have a pretty good ability to predict first hepatic metabolism. Again, this depends on intrinsic hepatic clearance, not CACO2. But now if we add the CACO2, we see that we tend to predict higher absorption than was actually observed. So there's a bias. It is actually correlated. This is still statistically significant, but it's not perfect. If we look at what escapes the gut, similarly, we're predicting, slightly over-predicting, but we do have a correlation. And then if we look at bioavailability overall, so that's the product of those three things that I showed you, there's a lot of scatter there, but there are actually, these are points on top of each other, and there are a lot of points on top of each other. This also ends up being statistically significant. How significant? Well, there's some p-values. Everybody wants p-values, right? So first off, when we look at the R-squared for HTTK without the CACO2, we were explaining 20% of the variance. Now, just to be clear, 20% of the variance is that you could win money in a casino with that. If it wasn't predictive, it would be 0. But that means 80% of the stuff that we don't know what's going on. So when we look at what we can do with the for the overall fraction bioavailable with CACO2 data, we have about 37% of the variance explained. That's okay. If we just look at the fraction absorbed, we're basically the same answer, 40%. And when we look at this CRISPR that we've developed, it's down to 30%, but that's still better than not using the CRISPR. And it's halfway between not doing it and using the CACO2 data. And unfortunately, given this audience, I was really hoping we had somebody here who was going to say, well, 37% R-squared is not good enough to replace an animal study. I don't know that, is anyone willing to raise their hand in the room? Because I'm going to make fun of you, but if you'd like to, just in full disclosure, because you sit there and you say, well, 37%, how can that be good enough? Well, let's look at what else we can do. So we've got ADMET Predictor, GastroPlus. I mean, this is basically a tool designed to predict the gut. We look how they do, and they really nail fraction gut. Actually, I should go back and point out, we really were terrible at predicting gut, about R-squared of 0.23. They're more like an R-squared of 0.5. And they're about the same, actually, for overall bioavailability, but they, actually, they're much worse for overall bioavailability. They're good for predicting gut metabolism, but overall, their training set, they're very focused on pharmaceuticals, and I'm giving them a bunch of non-pharmaceuticals, and so they actually do worse than HTTK without even considering CACO2 data. But I'm sure if I put a set of pharmaceuticals in, they'd wipe the floor with us. ADMET Predictor does really well with the gut, but it's doing a more sophisticated gut model. We discussed that yesterday. I think they have 20 different compartments modeling the gut, whereas we're getting away with one box for HTTK right now. But the real question is, what does a rat study do? If you've done an intravenous and an oral dose and you've contrasted the AUCs and you've done an animal study, well, there's a great paper actually from the SimCip folks, Muster et al. Back in 2014, and we find results pretty consistent with them. And there are more than 3 points here. There's a bunch on top of each other at high absorb. But the result you get from an animal study, so if I use rat, is about 0.2 R-squared to predict human bioavailability. So that's rat in vivo bioavailability has about the same R-squared as HTTK without having done any of this, and half the ability of CACO2 and lesser ability than the CRISPR. So my machine learning model beats animal studies, and actual in vivo in human relevant CACO2 wipes the floor with it. So that 0.37, yes, there's a lot we don't know, but relative to what a rat study gives us, we actually know a lot. And that's not a surprising thing. Our digestive tracts are very different from rats and mice and dogs and everything else. So the impact on risk prioritization, basically in the purple dots here, we have, again, the hazard converted to mg per kg per day. In yellow, we have the exposure. Because of the CRISPR and the in vitro data, we are now able to do basically 1,000 chemicals. And only these chemicals in orange at the left have this plausible idea of an overlap. And what the CACO2 data tends to do, because it says you'll need more food, more of the chemical, it tends to actually minimize the risk for some of these chemicals. But it hasn't made a huge impact because we were already talking about orders of magnitude. You know, if you were separated by 100 million, maybe you're separated by 1 billion now. You can get this data online. I'll share these slides. They're cleared. I won't go through all of this, but basically there's a variety of ways to get the numbers. You don't have to be a computer programmer to get these conversion factors. And so just to summarize, HTTK is an approach developed to address the gap of toxicokinetic data. We already had first pass hepatic metabolism. We have introduced CACO2, membrane permeability, from both literature chemicals and new data generated by the EPA. And we've built a machine learning model so that we can cover chemicals that don't have in vitro data yet. And when we put all these together, we are better able to predict kind of the margin between exposure and bioactive doses. And so this is a huge team that I'm honored to work with that's working on various aspects of HTTK, but for this project in particular, I need to thank Greg Honda and Elena Kenyon, who basically led a lot of the analysis, Chris Strock and Roger Donalo, who led the data generation, Barbara Wetmore, who keeps me grounded in, important things like, gravity's down, and everyone else on the project who's been a pleasure to work with. And so the references, these slides are cleared, so I'm happy to share them however you want them, and shoot me an email if you have any questions. I have no idea if I've left time for myself for questions now, but thank you.
Thanks, John. We do have time for a couple of questions. I thought I saw someone online raise their hand while you were speaking, but it's gone back down. So just as a reminder to attendees, either use the Q&A box to type your question, or please raise your hand to ask a question verbally.
Hi, John. Great presentation as always. And I'm so glad to know that once we get to a point with as we continue to improve the in vitro models in the laboratory, that the data can go somewhere useful. Can I get your perspective on, I agree with you, you're totally the high throughput guy, like right, yeah. John and high throughput, I totally get that. But the high throughput model that people are working on may or may not be accurate or precise for the endpoints that we're measuring. So can you speak a little bit about having a completely simplified, as simple as possible, Occam's razor, simple, suitable for high throughput, versus some of these middle ground of where we might need some sophistication with co-cultures or organoids or things that are not as high throughput, things that we can't do 1,000 or 10,000 chemicals with. So where's the place, is there a place for both? Well, what's your perspective for both types of NAMs?
Yeah, no, that's my favorite topic actually, because it's kind of, what it gets to is, can we, or my interpretation on it, when we're wrong, can we understand why we're wrong? So my favorite example is that there are, everything I showed you, there are no transporters modeled. Now there's transporters in the CACO2 assay, but I don't know what transporter was involved. But Alex Troch's group at UNC has QSARs that can say, oh, there's a 90% chance that your chemical is a substrate for this transporter. And if my data doesn't match, well, that's a chemical then that it makes sense to do you could do an in vitro transporter assay. That's not necessarily something that's affordable for 1,000 chemicals, but it's very affordable for 100 chemicals or 10 chemicals so that you have leads. And so I think we use the computer models to try to understand why we've gone wrong. And a class that's been difficult, for example, is PFAS. And Barbara Wetmore right now is leading a group of transporter assays for specific PFAS to say, okay, can we tweak for those particular chemicals by getting that in vitro measurement? And so you would I think you have to use models to kind of predict which in vitro assay is the next step.
It would be great if the slide that you have that kind of puts the IVIVC where you put where the in vitro PK or TK versus the in vitro PD and TD, if there can be like a box of where does the in vitro biochemistry go? Because like you said, like enzymatic activity is something we never talked about here, but there are tons of enzymes that are involved in the GI tract from the beginning, the saliva, all the way through the end. And so this community, we are getting to the point where there's going to be some lock and key mechanisms and wonderful enzymatic assays that are even cell-free that could potentially be high throughput, but that's like biochemistry stuff. And it would be great to figure out where that group of scientists could fit into your larger site.
No, that's a really good point. And we talked a little bit about the difference between just using the tier assay and actually measuring the permeability yesterday. So when we do in vitro measurements of plasma protein binding, one of the things that they do is they literally add the chemical to some plasma, and then they just assay that after 4 hours. They're not even looking for concentration, they're just looking for stability. And 10% of the time, maybe more, the plasma just chews up the chemical. You actually see that activity. We tend to not even report on those data because they're in supplemental table 45 or whatever, but we don't write a paper on it, even though in a way that is a very, very useful assay. Here's my list of leads for chemicals that would have some sort of enzymatic activity. We think that was a control in this one experiment, but that control is itself a useful measure.
Okay, thank you so much. We do have a question online, but I'm going to have you type the answer because we're going to move on in the interest of time. So now Kristen Gilchrist is going to speak to us about a three-layer intestinal model for toxin translocation studies.
All right, hello. I'm actually going to tag team this one with my collaborator, Angela Melton-Selsa. We're going to talk about a three-layer model that we use to look at Shiga toxin translocation. We're both from Uniformed Services University, which is just on the other side of Rockville Pike. That's the Department of Defense Health Sciences University. I'm in the Center for Biotechnology, also known as 4D BioCubed, and Angela's in microbiology and immunology. And you'll see their pictures at the end, but I do want to note that this is the work of a couple of postdocs who are no longer with our organization. Andrew Lamont was in my lab, and Rebecca Bobo was in Angela's lab. And so I'm going to do my best to represent their work. And these are just our own opinions. We're not speaking for USU or the Department of Defense. So why do we care about Shiga toxin? Well, in general, the Department of Defense cares a lot about diarrheal and intestinal illnesses because it's a real threat to, troops deployed all over the world and affects readiness. Shiga toxin is typically obtained from ingesting Shiga toxin producing E. coli from contaminated food or water. These stec infections can cause abdominal pain, diarrhea, and a non-insignificant number of cases lead to hemolytic uremic syndrome, which can cause kidney failure. There are two types of Shiga toxin, 1 and 2, and E. coli can produce either or both. And the Shiga toxins induce disease by traveling from the intestine into the bloodstream to localized sites like the kidney. Once you have hemolytic uremic syndrome, there's no really specific therapies. So the goal is that maybe if we understood more about how Shiga toxin was crossing the intestine epithelial barrier, then there may be more treatment approaches. So animal studies have not worked out as well. Like mice have to get a hugely high dose to detect Shiga toxin. So most of what is known is in vitro. And typically just monolayers of epithelial cells like Keiko, which we've talked about a lot. There's a variety of potential transport mechanisms. So transcellular transport's most commonly reported. There's conflicting evidence of micropenocytosis. There's been reports of paracellular transport with direct damage to the mucosa and barrier, and then also trafficking by immune cells. So in summary, there's been a lot of different methods presented, some conflicting evidence, and this may be due to variability in the in vitro models. There's evidence that the Shiga toxin behavior is modified in tissue models with greater physiological complexity than these monolayers, including some recent work with intestinal organoids that showed that having mesenchymal cells did change the epithelial barrier properties. So we kind of had that in mind with developing a 3-layer model in a transwell. So I'm an engineer. My job is to tell people that transwells are not good enough and that they need flow and stretch. So I'm losing my engineer card by showing a transwell model, but I think in reality, that's the way to efficiently see if this model was even any good for what we wanted to look at. And then we can always, refine and add those other things, flow, stretch, and including we had interest in adding a bacteria layer, but that kind of tends to overwhelm things. So we would need to look at, how to do that and have them live in harmony. So we do have a, endothelial and epithelial layer, and also an extracellular matrix containing myofibroblasts. More specifically, this is a primary cell model. With human cells, these were sourced commercially from Cell Biologics. So we have a 1 micron 4 PET membrane with a collagen gelatin matrix with the myofibroblasts, and then the primary epithelial seated on top of that. And then on the underside there, we have the colonic microvascular endothelial cells, also human primary. But what we want to know is, does this complexity make a difference, particularly since most of the literature was on monolayers? So we made a kind of single layer version with the same cell type. So same transwell, same epithelial cells, matrix without the myofibroblasts. So just looking at these cells and, if they were as expected, we did see mucin precursors in the epithelial cells. The fibroblasts did have alpha smooth muscle actin, so a mature marker of mature fibroblasts. And then we looked at the co-culture. So the myofibroblasts and the epithelial cells are on the same side of the transwell, so they need a common media. We did the fibroblasts, waited till they got mostly confluent, put the epithelial cells on top, found that a 3-to-1 mixture of the epithelial media and the myofibroblast differentiation media sustained viability for at least 2 weeks. In figure A there, you see they did consistently form these clusters, which maybe is start of some villa-like structure. And we could distinguish the kind of stretched morphology of the fibroblasts. And again, stained for the mucin and smooth muscle actin. So then for the tri-layer model, we started with the endothelial with the transwells reversed after 2 hours, then we flipped them into the fibroblasts. We waited 5 days then before doing the epithelial cells on top of those, and then waited 7 to 10 days before doing the experiment. So it's very hard to image all the layers on the transwells, particularly the undersides. We did some slices, and you can see the apical side, looks very nice with the 2 cell types. The endothelial side, there's definitely something there. We can't prove that it's confluent, though it's, it could just be slicing damage because it's such a thin layer, but we definitely have cells present a little bit more clear in fluorescence. So in terms of using this model to look at sugar toxin, I am going to hand off to Angela.
Thanks, Kristen. Yes, so Kristen's lab generously sent us the transwells to test, and we were really interested in this question because, as Kristen said, how sugar toxins translocate across the gut to get into the bloodstream and to target the kidney and the most serious forms of the disease is poorly understood. Another aspect that's poorly understood is that sugar toxin type 2 seems to be more potent in humans, even though these toxins have the same mode of action. They're around 60 to 70% homologous. And actually, SDX1, if you test it on viral cells, is more toxic to those kidney cells than is SDX2. But what we see in humans is essentially the opposite. So we were really interested in this. So what we first did was to look at the single cell layer model. And prior to testing in the transwells, we tested these primary colonic cells in monoculture for sensitivity to toxins, because we did detect very modest amounts of the toxin on those cells, which is GB3. But what we found is that these primary colonic epithelial cells were resistant to toxin, and so we proceeded to these transwell studies. And what you can see, and I don't know if this works. Yeah, okay. So if we put it Shiga toxin type 1, the prototype is 1A, so we always use the prototype in this, but we left the FTX1A there. And if we put it on the apical side and measured on the basolateral, or put it on the basolateral and then tested on the apical, we got toxin translocation, and it was basically the same. It didn't matter which side we put it on. And this is roughly 0.01% of the toxin input. I'm not even showing the input because it just throws the scale way off. For SPX2, we saw similar, statistically similar, and again, it didn't matter which side of the transwells we put the toxin on, we saw it go both ways. Now, one thing I was interested in these data about, even though in some ways they don't look that exciting, I mean, it's exciting, we got toxin to go across, but other researchers have seen toxin go across in cells that, like HTT8 cells, KCO2 cells, T84 cells. They've seen translocation of toxin. But in some of those models, they've seen differences between FTX1 and FTX2, and we didn't. But if you, from our lab, if you orally give toxin to an animal, we see the same amount of toxin going across the gut. So these data more reflected what we actually saw when we gave toxin orally to animals, which of course is not how they naturally get it. They would get it, or people get it, they would get it from the bug. But I was still intrigued by the fact that we got similar data. So when we moved to the more complicated triple layer model, we essentially saw the same thing. FTX1 goes from the apical to the basolateral, from the basolateral to the apical, and Shiga toxin type 2 does the same. And so we were kind of a little interested in this, but what was most intriguing about this, and I forgot to point it out on the previous slide, in the previous slide, what we saw as measuring of the toxicity dose as measured on Vero cells, we were getting about 10 to the 3 cytotoxic doses per mil for these toxins, but in the more complicated model, we were actually getting 10 to the 4th. And we had very few instances where we saw no toxicity or no toxin translocation, whereas in the single layer model, we did. So it's really intriguing to us that this more complicated model, we're actually seeing more toxin translocation. And so it may really be important to look at more complicated models, because in order to get the hemolytic urine sick syndrome, which is really due to the extreme kidney damage, you need toxin to cross that gut. So I think as we go forward, it will be really interesting to look at how the toxin is translocating across these kinds of layers. Okay, of course, though, people are infected with Shiga toxin producing E. coli. They aren't getting large doses of toxin, hopefully. So we did try some infection models. So we tried 2 different types of strains of Shiga toxin producing E. coli. The 157H7 is responsible for very large outbreaks that you've heard about in the United States, like from hamburgers or things like that. We also tried an 026H11. This caused a Chipotle outbreak, for example, not this particular 026, but that's a serotype that has caused outbreaks in the United States as well as other countries. The difference between these 2 strains, besides the serotype, is that the 0157 strain only makes SPX2. The 026H11 makes both toxins, and then both of these toxins have the gene for intimin, which is called EAE, and this intimate adherence factor caused really tight intimate adherence to the gut epithelium. So we thought it was important because the most virulent strains of SPX2 have the intimin. So when we infected the model for 6 hours, we didn't observe any toxin translocation at 6 hours. So we extended the time period, and our 0157H7 strain destroyed our model. In fact, we saw bacteria on the other side, and we saw 50 on the other side. We're adding 50 to the top. And when we knocked out the adhesion, we still saw destruction of the model. So I just have a very little bit of data from the 026H11 strain, which didn't destroy the model in that time period. And what we saw from infection, looking at CD50s per mil. In the basolateral side, we saw 100 CD50s per mil, so low levels of toxin crossing from the bug. And if we made a mutant in Shiga toxin type 2, looking at the green, we couldn't detect any toxin on the other side. And we were unable to make an STX1 mutant in this strain. So what we did is we added anti-STX1 antibody to essentially mimic an STX1 mutant. And for that strain, in that condition, we also didn't see any toxin translocating across. So I think going forward, it will be interesting to do some additional types of these studies to see how bacteria can actually translocate toxin, but we may have some limitations about which types of strains we can use. And I think we heard some really interesting models yesterday. There may be some modifications that we could make or some different models that we can try. And one thing I didn't mention, based on the talks that I heard yesterday, some of these toxins are modified in the gut, particularly Shiga toxin type 2D that I work with. And so there's a lactase in the gut that can cleave that molecule and increase its potency. In fact, what it does is it then binds to more receptors in the gut. And so if we could model that in this kind of system, I would really be interested in that kind of thing. And then, so in summary, we developed the two models. We saw similar translocation, but the more complicated model, we did see increased toxin translocation. We're very interested in that. And so we want to further our understanding of that model. And I just put a ribbon diagram of the crystal structure of Shiga toxin over here, because it's kind of pretty when we're looking at it, not pretty in our bodies, of course. And then lastly, these are the postdocs that were involved in these studies. And Kristen and I would be happy to entertain any questions.
Okay, thanks very much for that presentation. I'm not seeing any questions in the Q&A chatting. Christy?
Yes, so I don't think we've heard a lot about these myofibroblasts yet. So what are their role? And they're embedded in a matrigel kind of thing. And then secondly, did you do translocation studies with just the Keiko cells without that matrigel layer? Like for instance, in a normal plastic polystyrene transwell, did you put just do just Keikos without that myofibroblast layer?
So we didn't do the Keikos, but we did the primary epithelial cells, and that's the first data that I showed you. Correct. The very first one that I showed you, where both toxins go both ways, but it's less toxin. Other groups have done Keiko-2 cells as well and seen translocation.
So other than, I mean, this finding that you actually see more translocation, what is the role of those myofibroblasts, either in the body or in the system? What were you trying to recapitulate there?
I'm sorry, I actually read what they do, but I'm sure that I would misstate it. They have a lot of roles in the intestine, but I'm sorry, you know, I'm gonna play the engineer card again. So yeah, it is. Right. Yeah, it is there.
It's underneath, right, those cells. I'm sorry, we should have had our postdoc here to explain why they did. But they wanted those cells that were underneath, that would be underneath possibly the epithelial cells. Right.
Yeah, and I think part of the inspiration was this organoid paper where they showed that it made a difference. So that's why we felt like we should capture it.
So I don't have an answer for this, but I mean, I think that sometimes we're left with an overly functional perspective on these models. So the tissues are complex, and cells exist in a given environment, and that probably matters. I mean, normally we think about when we think about permeability, we think about the interface layer that's fundamental, and then we tend to ignore whatever is down. Yeah, so I mean, maybe there's some sort of entourage effect that may make a difference in modulating the way how the epithelial cells behave. So anyway, I think it's a really interesting finding that this is making a difference, and sometimes it's these serendipitous observations that can make a difference in the future. So, of course, I mean, that also brings challenges because, I mean, sometimes we recognize that we want to have the simplest model possible, and simplest is not always necessarily the best. So, anyway, really interesting work. Thank you.
Okay, next up we have Dr. Bill Thielen, from the Chief Scientific Officer from Altus Biosystems, to talk about an assay they have in their organization.
So thank you all very much for having me to come talk today. I'm here representing Altus Biosystems. So we are a contract research organization in Research Triangle Park, North Carolina. And just to give you a little bit of background on us, so we were founded by Nancy Albritton and Scott Magnus while they were at UNC. Nancy's currently the chair of engineering, dean of engineering at University of Washington in Seattle. And Scott's lab is a stem cell biology lab. So it's really a collaboration between engineers and biologists. At Altus, so we're still a relatively small company. We are 20 employees or so right now. About half of our company are PhDs, and we have 3 dedicated teams. It'll sort of reflect the things I go through in our talk today. So one is based on cell operations. So one of our goals at Altus is really to bring primary human intestinal cells, and make them commercially available, highly quality controlled. It's sort of the boring part of the talk and stuff, but it's really fundamental to this field. But we provide cells as well as in the forms of kits, as well as commercial services, so assays that we'll run with clients as on a contract basis. So what I'm going to focus our talk on today is basically on generating commercial scale primary cell culture models and making them available, and then how we quality control them and make these systems reproducible, and things that we can do lots of experiments on and not change around the cell models. And then I'm going to dive into 2 examples of assay development. And again, we're starting with fairly simple models. So these are transwell-based models that are these primary human GI cells. And just sort of our logic in developing these more simple models from the ground up in a fit for purpose way. I definitely want to touch on a couple aspects of model complexity and sort of matching the scale to the throughput requirement for the assay, and then touch on a little bit towards qualification and validation of these types of assays. So we start at Altus with full-length human donor intestine as the source of our intestinal stem cells. So we get in full-length tissue. It's from healthy transplant-grade donors. We isolate the crypts from each major region of the small and large intestine. So we have the duodenum, jejunum, ileum, ascending, transverse, and descending colon. We also get myofibroblasts from this tissue. As we go forward with additional tissue isolations, we're also working on getting immune cells, gall tissue, endothelium, microbiome, things like that, so that we can have immune matched tissues with all the different cell types represented. We do have commercial donors that represent male, female, different ethnicities, different age ranges, BMI. So we are working towards building out a genetically diverse pool of donor tissue. And as I'll show you in some detail, what we do from there is expand those stem cells so that we can make commercial scale lots. So it turns out for 1 vial of crypts that we get, and we typically get hundreds from each region and still don't use most of the tissue, we're able to generate tens of thousands potentially of cells scaled up from there. And I will go through a bit of our quality control, because it's really important that we don't see genetic drift or phenotypic drift in the cell populations as we expand them to make them scalable for use in a number of different assays. So essentially what our model does, and again, this is going to speak through our replicate system, we call it, but this is a transwell-based assay. It reprises the entire life cycle of the native GI tract. So starting with the crypt base. So this is where you have your stem and transient amplifying progenitor cells. So these are the cells that you will have that divide and constantly allow the gut to renew about every week. The cells will grow up and differentiate into the diverse lineages like goblet cells, absorptive enterocytes, and enteroendocrine cells. So we isolate the crypts and we expand the cells on a two-dimensional plane. So a lot of the early work of the company went into how to do that successfully and maintain the phenotype and the genetic stability of the cells. And then we're able to biobank them. So we can freeze them at that point where they're essentially ready to use. So the way that we provide these, you can thaw the vials, it goes straight onto the transwell. I'll go through that culture system on the next slide, but we went with the transwell system. It does offer some advantages to organoids. There's sort of pluses and minuses on each system, but we did like the access to the apical and basal compartments. So it allows us to do electrical measures like TIR on the culture system, as well as do bilateral sampling for drug transport studies. So to just sort of walk you through what our cell model looks like. So when you thaw a vial of cells, plate them on coated transwells. The first 5 days shown in the schematic are dominated by the proliferating stem cells of the gut. So we show an image in the bottom, which EDU staining. So it's basically just labeling the proliferative cell population. On about day 5, they reach confluence, at which point we change the media. So we change from a media that supports stemness to one that will support differentiation in the cultures. It takes the cultures a couple days to fully differentiate. When they make contact, they do start forming electrical resistance. And within a couple days, they'll differentiate into the different cell populations, again, the enterocytes, enteroendocrine and goblet cell populations we show stained on the sort of bottom right panel. An interesting aspect of the culture. So, we use TIR fairly heavily as a measure. We've heard a lot about this measure already. But the TIR profiles for the cultures are different for the different regions of the intestine. So with the transverse colon, for example, you see it establishes a high electrical resistance very quickly in culture. It largely plateaus, and then you see the large drop off of that. So that's the actual normal apoptosis of the gut. So it's the normal life cycle of the intestinal epithelium. So this is once the cells are starting to differentiate, we have somewhere between 5 to 7, 8 days to study the cultures. The small intestinal cultures actually have a very different profile. So they take many more days to actually reach what we would call a peak TIR value. Which we think actually reflects the fact that they typically have both a crypt and a villus to sort of migrate through, but this is common for small versus large intestine cultures. So one of the things that we did very early on as well was to ensure that we had genetic and phenotypic stability with the cultures and understanding how we could expand them and commercially scale them. So we ran a whole series of studies. So just a couple examples of the types of data we looked at. We looked at gene expression changes over time with passage. We have looked at chromosomal stability in the past, but we also looked at things like the TIR profile. So on the graph on the left shows that we compared cells at passage 2, 5, 10, and 15. That as you get into higher passage numbers, especially 15, it becomes very obvious that we're seeing a lot of these different sort of genes. A lot of these are markers for enterocytes or goblet cells, and things start changing fundamentally. We see changes in metabolic enzyme expression. So we use this type of data to fix our passage number for each region of the gut. And when we look at data like the TIR data, you can see a very clear difference in the TIR profile with the cells between passage 5, 10, and then at passage 15, it fundamentally changes and becomes more variable. So we used all of this data to basically lock passage numbers. It is different for each region of the gut. Some are more sensitive than others, but we use that as the sort of starting point for scaling up cells and then performing quality control assays. So again, to go through a TIR profile and how we assess quality control on all the cell lots we produce. the way that we commercially produce these, we'll take a vial of Crips and expand that into somewhere between 200 to 400 vials that would constitute a single lot of cells. Those are things that we could provide. We have clients that are interested in owning an entire lot, or certainly owning, being able to go back to the same donor, same region each time they do a study through multiple lots. But we run a fairly extensive quality control where we look at parameters like, initially things like cell count and viability. We look at sterility, but then we do functional assays on the tier profile. So just following the schematic on the left side, we look at time to confluence with the cells when they start producing a barrier. We look at time to, we call it peak tier, so generally about day 2 for the transverse colon. We look at what we call the tier plateau, so we look at the starting day to the plateau length, and analyze all of these parameters and have set quality metrics around them. So the graphs on the right-hand side show this is now for data that is our QC metrics averaged over 50 independent experiments across 4 tissue donors that have diverse demographics. You can see that on day 0, when at confluence, essentially, we have a very low tier. It becomes much higher. You can see the plateau. So we set metrics around what % of that peak tier the cells maintain for a given number of days as sort of a critical quality metric. We use the coefficient of variance across that, which is the bottom graph. So we set that metric at being below 25% to pass a lot. So you can see on that graphic that as you get into day 2, 4, this label day 2, 3, 4, 5, and those are the days with the lowest variance. So that's where the tier is fairly stable on those cultures. By the time you get to day 6, the cells are starting to apoptose, so we see the variability coming into the culture. But it helps us define a really nice sort of fixed assay day for the cell system. And so we issue certificate analyses with all of the data on every cell lot that we sell for these. So I'm going to now switch into developing some different assay systems using this. And I know from all of the talks we've heard earlier that this group doesn't need a lot of introduction to GI tox models, but certainly, we started there because, it is clearly recognized that GI toxicity or adverse events in clinical trials are the most frequently reported for any target organ across all drugs that go into clinical studies. And it's certainly a very good example in which the non-clinical species have not been highly predictive of this. So, there are a lot of really nice meta-analyses of these types of data. But in one example, rat and dog across clinical trials is only found to decrease or it's a reduction in certainty, but basically, only 20 to 30% of the time do they help inform whether a human is going to have a GI adverse event in a clinical trial. The non-human primate is much better, but with ethical considerations, cost, and very, very low throughput, that's usually reserved for only sort of lead compounds or near clinical stage drugs to be tested. And the problem with this is that what this leads to is our drugs going into the clinic and then causing these GI adverse events that weren't predicted by the non-clinical studies. And sometimes they can be severe and life-threatening, or certainly they can be problematic for compliance and things with the medication. So we sought to use our model to look at this more. And you've heard some nice talks already on some of the in vitro models with organoids from Julie yesterday. So in our, I want to cover in our transwell-based assay, one of the things that we're doing is we're taking advantage of the dynamic culture conditions that we have in our system to really focus on how do we build out a robust but also fairly high throughput assay. So this is a 96-well plate-based assay system we run. It is a 5-day assay end-to-end. So we start with basically thawing vials of our cryopreserved cells, plating them on transwells, so they'll sit down and be somewhere between 40 and 50% confluent from the start. So over the By day 3, they've reached about 80% confluence. At this point, we're dosing the compound. So one of the things that we found, and I'll touch on, is that often the pharmaceuticals that we're often testing on these models will target proliferative cell populations more so than some of the non-dividing and differentiated cell populations. But we can assay both on this. And then by day 5, the cells have reached confluence. We measure TIR because they've established an electrical barrier. We also label the cells with EDU during the process to label and stain differentially with DAPI to quantitate the difference between the number of cells that are proliferative versus the number of cells that we have there in total. So with a fairly straightforward assay, again, we can run multiple 96-well plates at once on this type of experiment, 5 days, so no weekend time on this one. We can get barrier formation, we can get cell viability, and we can get an index of cell proliferation. And just to show you where that sort of gets interesting with some compounds, so I'll show you some of the compounds we've tested on the model already. So idarubicin is a DNA intercalator and topoisomerase inhibitor. So it's an anti-proliferative. It's a chemotherapy drug. And if you look at the dose responses, which we can run these readily because of the 96-well plate format of this, you can see that if we look at the change in cell proliferation, so in the green line, that drops. That's the sort of most sensitive metric. TIR sort of follows, and then the total cell number viability is the least sensitive metric on this. So we get sort of multiple shots on goal towards what's actually happening to the cells. So this makes sense, and the anti-proliferative is going to grow the rest of the cells that will ultimately impact the barrier function, and at a high enough dose will basically kill all of the cells. But it gives us a very sensitive way to interpret these data, and I actually show the clinical Cmax for idarubicin, overlies quite nicely with the proliferative measure. So we're predicting toxicology at roughly the same concentration that idarubicin is given and measured in plasma patients that are, in this case, have a fairly high frequency of GI side effects associated with taking that drug. So to give some other examples where we can use these measures to get some insights into mechanisms on the front end. So this is basically 2 examples of a drug that is non-specific. So and these are all the top panels is bortezomib. So it's a 26S proteasome inhibitor. So it's going to be toxic to any drug that is metabolically active, whether they're proliferating or they're differentiated. And the top left panel shows TIR. And in this case, we ran the assay on the stem cell population. We also ran this a second time where we let the cells go on to fully differentiate before we dose the drug. And you can see that TIR drops at about the same concentration in both the proliferative and the differentiated cell populations. If we look at or in those populations, if we actually stain for proliferation in total cells, you can see that bortezomib causes the complete loss of the proliferative cell signal as well as the DAPI signal, which is, again, just a metric of total cells still in the plate. So it's non-specific to both types of cell populations. But again, if we come in with something like an anti-proliferative, so in this case, this is colchicine, so it's a spindle microtubule disruptor. We can see that with the TIR on the left side in the red graph that the drug is fairly toxic to the proliferative cell populations. But if we allow the cells to differentiate, we actually don't assess toxicity on the cells. So they're tolerant of that drug. So that would represent aspects of the mature gut. When we look in our proliferative cell assay and we look at the staining of the proliferative versus the total cells, you can see that the green signal is completely lost in the presence of colchicine. So it's basically wiping out the proliferative cell populations, but the DAPI signal is not gone. It becomes patchy. So if you quantitate that, the graph on the right, you can see that basically as we increase the dose of colchicine, we lose the proliferative cell populations, but we do not touch the cell populations that were non-proliferative. So it does give us some insights into how we're targeting different cell populations as a function of the dose with those very simple measurements. And again, Julie gave a really nice talk yesterday with a lot of compounds that she referred to as the IQ reference compounds. So we have been running those compounds across our model. We still have a little ways to go, but just to show sort of a basic summary, all the compounds in sort of pinkish red are known to cause clinical diarrhea. We know what the clinical Cmax for those compounds are, and we can benchmark that against the IC15 or IC50 for our in vitro assays against those clinical exposures. And with all the positives, we can show that we're within tenfold or less of the clinical Cmax and the concentrations that are toxic in our model. We are also including negative controls in this model, so we show a couple examples in blue, but we continue to work through a much larger data set to sort of establish that model. So then to switch, so one of the nice things about the platform is that you can use the same cells on the same platform to run other types of assays as well. So this is actually a little bit different in that we're using the jejunum for this. We use transverse colon on the toxicity slides, but the small intestine is the major site of drug absorption in the body. And again, because we have the transwell system, we can take advantage of being able to add drugs apically or basolaterally and measure absorption or efflux through the epithelium and sample over time. To start establishing a DMPK model on our system, we started with some transcriptomic analyses. One of the advantages of having a founder that is the head of the transcriptomic core at University of North Carolina, they started with an entire data set from native human gut. So we have the single cell RNA-seq data for that, and we can make comparisons back to the transcriptomic data that we have in our culture set. So this is just a quick survey of a lot of relevant DMPK, drug transporters, metabolic enzymes, and expression in our system. I put a red box around two examples. So one is CES1 and 2, so these are carboxyesterases. I will actually show you some functional sort of examples that are sort of cool with that, but you can see that our cultures do not express CES1 as the small intestine should not. They do express CES2, but you can see genes like CYP3A4 is the bottom one highlighted. That this is the expression over time in culture, so that as the cells differentiate, we can see that gene becoming highly expressed in the cultures over time. So it was to give us some background on what type of metabolic activity and transport activity we would expect from our tissues. So we have gone on to do some functional testing of this. We're doing this in collaboration with Genentech. So Eugene Chen is the heads of their pharmacokinetic group. So they used our model in-house with, again, the same jejunum cultures and looked at initially just intestinal permeability with 24 pharmaceuticals. And these were all ones that we had published references for the human bioavailability measured as PF. And we compared that to the PAP that we got back for these compounds. So as a starting point, it's giving us a nice reference data set to understand the capacity of our cultures to transport drugs through them. Overall, we've gotten a fairly nice correlation through the data of the first 24 compounds. We have gone on to do a number of more detailed transport studies. So we have looked at molecule transport through PGP and BCRP, so we can see that drugs like digoxin and belastine, rosuvastatin and things are certainly effluxed in the BDA direction to those transporters. We've done that in the presence of absence of inhibitors. Some of the data that we're not going to show includes other transport processes through channels like PEP-T1, which tend not to be expressed in CACO2s that are more of a model of the colon than they are the small intestine. And we've also started doing a number of studies on phase 1 and phase 2 metabolic enzymes. So to go one step further, so all of this work was really about generating sort of a core understanding of the sort of simple base model of the system, we are exploring more complex models. So once you have the quality of the cells and reproducibility of the cells in the simple model, it's allowed us to look at more complex systems like we have done here with CN Bio. We're using their physiomimic system to join our small intestine cultures with their human liver model to generate a gut liver model that we are hoping to be one of the sort of early versions of a true sort of human oral bioavailability predictor, because it would include first pass metabolism in the model. Essentially, the way this works is that we have separated compartments for the intestine that the basal compartment flows over the liver. I will say it was a fair bit of work to work out the media conditions that would support both cell populations, but we got that nicely optimized. But we can apply drugs to the apical surface of the intestinal epithelium, watch it either be metabolized or transported through the gut, and then circulate over the liver, and we can look at what either passes through the liver or certainly hepatic metabolism. So one of the compounds that we ran as a proof of concept was temacaprol. So temacaprol is a prodrug, so it's masking carboxylic acid with a short carbon extension that makes the drug permeate through the GI tract. So one of the challenges with this, and historically KCO2 models, to look at this, and one of the enzymes that I mentioned from the transcriptomic data, temacaprol is metabolized typically in the liver by the CES1 enzyme. And looking at the panel there, this is a Western blot of expression. So the small intestine should not express that. It is not expressed in our cultures, but KCO2s express CES1. The small intestine should express CES2, but that does not metabolize that compound. And so what we can see is if we apply temacaprol to the apical surface of our cultures versus an equivalent gut liver model with KCO2s, the KCOs extensively metabolize the temacaprol early on, and we actually see that drug pass through unchanged and through the epithelium, then to go on to be metabolized in the liver, which is shown in the bottom panels. But it's just sort of one start of trying to build out these systems, and something that we look forward to exploring more in the future. So I'd like to thank certainly the NIH, who has supported a lot of this work, both at NIDCR and NIEHS, our industry collaborators with Genentech, Roche, CN Bio. We didn't talk about studies with AstraZeneca, and as well as our academic founders and the Altus team. Thank you.
Okay. Yeah, that's all right. He's got a microphone over there. He can answer questions if you'd like. Any questions in the room? Okay, so I do see Patrick has his hand raised online. Go ahead, Patrick.
Thank you, and thank you, Bill, for a very nice talk. I was a bit surprised that you said that PEPT1 is not expressed by CACO2 cells, or did I misunderstand you? We find PEPT1 at the protein level, and we can also demonstrate its activity in CACO2 cells. And I think that's been known for quite some time.
Didn't hear the question.
Sorry, Patrick, we're having a little trouble understanding you.
Sorry, I will speak more clearly into the microphone. Did I misunderstand you, or did you say that CACO2 cells do not express PEPT1? Because we have good evidence that they do, both on the functional and protein level.
So we didn't look at that specifically, although there's a lot of literature reports that KCO2s, the sort of the channel systems that they are, the transporters that they don't express well in a lot of labs' hands are PEPT1 and certainly aminoglycoside transporters, but certainly may be the case that they have it.
Okay, could be. Thank you.
I have a question, Bill. I think I know the answer based on conversations yesterday evening, but you mentioned the access to diverse populations, and I just wonder what Altus has in the works associated with a model system that could capture population diversity and perhaps susceptible populations.
So we are continuing to build out our donor bank, and you know, with respect to donor diversity, you know, we are, and it's fairly common for us to screen, you know, on these types of platforms. It allows us to screen across multiple donors in a fairly efficient way. So I think over time, that's certainly a piece that's important to us, as well as having all of the different regions of the gut to screen across as well, as opposed to just focusing on a single region.
So, I mean, actually related to this is a lot. So my understanding is that most of the absorption of toxicants will occur in the small intestine, and yet we are developing models that are largely based on cells from the colon. And I don't really know enough about differences that are to be expected between cells from the colon or small intestine, but are they meaningful?
You know, so I will certainly say that, I mean, we know from the single cell transcriptomic data that the enterocytes in the small intestine are functionally different than what's in the colon. And one example that we've worked on in collaboration with our founder, Scott Magnus. So the small intestine, you know, has a tremendous capacity to absorb and metabolize fatty acids, which a lot of toxicants may be actually hitching onto that pathway. So we started looking at that. So this would lead to chylomicron secretion and delivery to the lymphatic system versus the liver and the portal vein. And we've started working on a model system that would recapitulate that as well. So, I mean, it becomes important to have all of those processes, but certainly, I mean, fatty acid is one great example that anything that binds fatty acid binding proteins or intercalates into lipids might actually be trafficking the GI epithelium by that pathway, but that is not reprised in the colon.
Exactly, and so that aligns with what we were chatting about yesterday in the afternoon about the fact that the matrix that makes it to the GI tract can make a difference in the absorption of some drugs. So that's all aligned with it. And Bill, by the way, I mean, congratulations. That's really the right path, trying to establish some sort of biobank of cells that can represent human diversity in the human population. That's really important.
You know, it's an important starting point because ultimately the cells can be agnostic to the platforms that you want to put them on or the experimental models, but you need high quality starting material as the first step.
Thank you, Bill.
Yeah, Bill, building on what Gonzalo just said, I almost wonder, especially with the CN biosystem where you're linking two organs together, I wonder if it's worth doing some studies on the small and duodenum system, and then thinking of the colon as a second or completely different organ and putting them like that to maybe, yeah. Are you doing that?
Love the idea of doing that.
Yeah.
You know, we're again sort of biting all of this off sort of one step at a time. So, you know, given the role of the small intestine in you know, absorption for the GI tract, and it seemed like a relevant place to start. We are following up with a colon hepatic model as part of our R&D collaboration, but you know, it'd be fascinating one day to link multiple chips and simulate sort of transit through the entire GI tract. But certainly we have the cellular material to be able to do that. It just becomes a technical exercise over time.
True. Okay, thanks again, Bill. Why don't we take about a 9-minute break and back here at 10:55. Okay, so our next speaker is Dr. Patrick Lundquist from the Department of Pharmacy at Uppsala University. He's going to talk to us about a human stem cell derived intestinal organoid system. Patrick.
Thank you. Thank you for inviting me to this talk. Let's see if we can. So if this is showed in the correct manner now.
Yes, that's correct.
Yes. Excellent. Thank you. And it's been a very pleasant 2 days. One of the good things about being at the end is that there's so much of the groundwork has already been laid. So I realized that a lot of my background is totally superfluous by now. But let's thank those who made the lion's share of this work. That is our PhD student Rebecca Hammar, our PhD student Mary Ceylan, and Daisy Hjelmqvist, a very skilled researcher. And that's our little team. And what we have been focusing on for a very long time is ADME and in vitro ADME especially. And as you all know, this is a large part of the pharmaceutical industry, where we try to get good pharmacokinetic properties of new drug candidates and de-risk them, etc. But then in S&M, we can apply the same methods to get exposure-based risk assessment and toxicokinetics, as has been talked about several times during these days. And what we are working mainly is the absorption and metabolism part of this. We have some small projects in the kidney as well, and we're also looking at the distribution into organs and cells. But to a very large part, we are looking at absorption and metabolism in the intestine and liver metabolism and transport. And the concentration curve in the patient, of course, or the person exposed to an environmental chemical will depend on these properties. So delineating or influencing the absorption phase, the distribution phase, and then eventually the elimination phase. And together then giving the AUC value under the curve. And a central factor in this is then the oral bioavailability. How much of a given oral dose is absorbed and escapes pre-systemic and first pass metabolism in the liver to reach the systemic circulation. And that's been mentioned several times these days. The animal bioavailability is often a very poor predictor of human bioavailability. It's more of a scatter plot than anything else, which means that we need new and better tools to look into this. So what we are doing then is as a combination of biologists and engineers and pharmacists, we start with the compound properties, working with several different modalities. Nowadays, biologics, small molecule drugs, peptide drugs, etc. And then looking at classic and new in vitro systems to study these. We often try to characterize these with global proteomics to get an overview of the protein expression levels in our models, and then comparing that to clinical samples that we get from the local university hospital. We have rather good access to especially liver and intestinal tissues, which is a big advantage, of course. And knowing then the different kinetic constants that we can generate in vitro and the expression profiles of our models and the human tissues, we then try to build physiology-based pharmacokinetic models, or in this case, then toxicokinetic models, as we heard earlier today. And a mainstay of this is, of course, the CACO2 cell model that we've talked about a lot during these days. And we use it in a rather basic setup, single culture on normal tense well filters, looking at directional and bidirectional transport. We have characterized the cell line extensively when it comes to uptake, efflux transporters, the different metabolic enzymes that the cell line still possesses, which are usually not the main ADME metabolic enzymes, but we have carboxyesterases, for example. We have a lot of brush border proteases and peptidases that present a real barrier to peptide drugs, for example. And using this, the permeability rate constant we can get from this model, we can then predict the fraction absorbed in the normal manner. Then we are also developing this model further. We looking into the mechanism of permeation. The question asked is what distinguishes cellular uptake, permeation over one cell membrane, from transepithelial permeability, where you have permeability over two consecutive membranes. And doing this, we've done a lot of mechanistic studies, and we can see that for many, many drugs, not surprisingly, we have a large accumulation in the cell layer of the CACO2 cells. And this is an important factor, but what seems to be the most important factor for the permeability mechanism is that the intercellular free concentration is sufficiently high to allow permeation out of the basolateral membrane. While the cellular uptake, of course, is driven by a concentration gradient over the apical membrane from the donor chamber and into the cell, this then has to create a sufficiently large intercellular free concentration for permeation over the basolateral membrane to take place. And these are questions that we are working on at the moment. And then in the project Risk Hunter, which is a EU-funded Horizon 2020 project, looking at exposure-based risk assessment and the use of in vitro NAMs to generate that, we've also started to isolate human jejunal enterocytes. And these are cells isolated from the mucosa of gastric bypass patients. We get a little intestinal section of about 2 centimeters. But this is enough to generate around 100 million cells with a good viability, 80% or above, commonly above 90%. And this is an especially fine example of a morphology. They don't often look as good and columnar as this, especially the cleanup step with gradient centrifugation seems to produce a sort of an osmotic stress on the cells, which makes them round up, but they still seem to be active and viable for quite some time. We can see that they present around 20% present with early apoptosis and around half that again with late apoptosis markers. If you use these for metabolic stability experiments for a few hours, these figures increase, much like they do in hepatocytes that are only viable in suspension culture for a few hours. We think that we can use these for approximately 4 hours in suspension culture. And one question is, of course, if we can eventually cryopreserve them, but we have not gotten to that stage yet. But we can see that they express a lot of the expected metabolic enzymes, both for phase 1 and 2. And we can see high activity of several, CYP3A4 in particular, CYP2D6, 2C9 as well. And then for some compounds, for example, propylparaben, we seem to have a large esterase activity as well. And when I say large, we have compared this to the hepatic levels, and hepatocyte activity is much higher than this, several-fold higher. But we see decent and measurable activity. So combining these with the CACO2 cell permeability, we also get a handle on the perisystemic metabolism, and we believe that this will improve our kinetic models. But then, of course, the dream would be to have a system which expresses both metabolic enzymes, the transporters we need, and presents with a permeability barrier that is at least reminiscent of the actual human intestine. And to do this, we have started to, as many others have talked about, start to produce stem cell lines, which we can use to produce organoids from different parts of the small intestine. At the moment, we're working on colonoids from the sigmoideum of the colon and small intestinal jejunal organoids that we call enteroids. And these are again then isolated from gastric bypass patients, while the colon tissue derives from either metastatic liver cancer patients or from biopsies from patients undergoing colonoscopy for diagnostic purposes. In that case, only the ones that presents without the diagnosis are used. And as Dr. Ko talked about yesterday, we can do these in different configurations. We can have a basic configuration where we have a basal membrane facing outwards and an apical enclosed space in the middle of a little ball. Removing the extracellular matrix, we can flip this to a apical out configuration, which is the one we most commonly use for experiments. And our hope, of course, is that we should be able to use these eventually then for transport and permeability studies. Yeah, and as many others have mentioned, we isolate the crypts, we isolate the stem cell niche from the crypts, we expand these and freeze the lines. And in a transfer to to a suspension culture, we can choose which orientation we like of these organoids. And this is actually quite efficient. We've tried to quantify it, and it seems like 97 to 98% of all the enteroids present with the wished for orientation. So if we, there seems to be a bit of a lag when I move the pointer. You can see here in the blue bar, in a sample where we have removed the matrigel, approximately 98% of the enteroids will then switch to a apical out orientation, while in a suspension culture with the presence of 7.5% matrigel, 97-98% will remain in the basal out configuration. And to the left, you can see a photograph of a apical out enteroid. This is not the best photo we have, but I think it's interesting from one aspect. And if you see the dark lacuna at the bottom of the enteroid. This is very reminiscent of how a goblet cell presents in a similar staining of a intestinal section from the small intestine with this unstained lacuna and a cellular nuclei that has been sort of shoved to the side. So even when we have culture in conditions which favor the formation of absorptive enterocytes, we still see a subpopulation of goblet cells. This is a bright field image, and you can see that they present in decently uniform size. Enteroids do grow more regularly than the colonoids do, and seem to be forming or wishing to form villus-like structures from time to time. But we are working on getting these even more uniform. Now looking at with the scanning electron microscopy, we can see that what look from the immunofluorescence staining looks like a brush border membrane. Also at the morphological level seems to be a true brush border with microvilli presenting all over the enteroid. It's of course nice to have a decently in vivo-like morphology, micromorphology in this case. And these early observations of goblet cell-like structures in the cell made us stain for MUC2 production, which we, for example, cannot see in CAC2 cells. And it turns out that a subpopulation of cells in these enteroids indeed do produce MUC2. The question is then, do they secrete this? Do we form some kind of mucus layer around the enteroids? That remains to be demonstrated. And we can also see that these enteroids form a polarized barrier. To the far left, you can see the uptake of a fluorescently labeled fatty acid in apical out enteroids. Presenting the same fluorescently labeled fatty acid to basal out enteroids generates no uptake. So we have a polarized expression of these fatty acid transporter. As you can see the quantification in the middle. And to the right, you see in green the expression of a fatty acid binding protein, which is also clearly localized to the apical brush border membrane, ready to receive imported fatty acids. And we can see a similar polarization of, for example, BCRP and PGP, and we are doing preliminary experiments to see the activity of these transporters, and the initial results seems very promising. We seem to be able to demonstrate BCRP activity, and I hope to be able to show that data in the near future. And then looking into the metabolism, we are now characterizing this with a rather wide panel, but we can for CYP3A4, which is the most highly expressed of the CYP enzymes that we can see in the enteroids, we easily follow metabolite formation from both midazolam and terfenadine. So this is 1-hydroxymidazolam and fexofenadine being formed by the enteroids. And you can also see that we can measure the loss of the parent. So we can do a classical metabolic stability assay here and calculate an intrinsic lint value for these compounds. And as I said, we are proceeding with cocktail measurements for a large selection of CYP and phase 2 enzymes, as well as carboxyesterases. We like to do especially proteomic investigations of our model systems. Very many of these membrane transporters that we're most interested in show a rather weak correlation between the transcriptome and the proteome. So here, in a collaboration with our colleagues in Signatop in Germany, we have looked into many of, and this is targeted proteomics of many ADME-relevant ABC, SLC, phase 1 and phase 2 enzymes. And as you can see on the far right here, we have in the enteroids as well as in the tissue, we have decently high levels of the expression of this enzyme in the tissue, in the isolated enterocytes, in the apical out enteroids, and in the basal out enteroids. And looking at ABC transporters, this is particularly interesting because especially PDP, but also MRP2 here, seems to be particularly highly expressed in the apical out enteroids, much higher than in the basal out enteroids. So this shift in orientation seems to affect the expression of these brush border located efflux transporters. And exactly what's behind this, we don't know at present, but it could be that this, in the basal out, we have a much smaller volume inside the enteroid, and perhaps this leads to an accumulation of compound that is not then conductive to the high expression of the transporters, but these are then upregulated in a apical out configuration. We can also see several of the SLC transporters that are important. SLC2B1 here is interesting. We see in vivo-like levels of expression in the enteroids, and also CACO2 cells, and especially our clone of CACO2, are known to express very high levels of SLC2B1, VOATB2B1. So these organoids are much more in vivo-like in that respect. And we can also see a downregulation of the, especially the SULs. They have lower expression levels in the enteroids, but it should still be clearly measured. The activity should hopefully still be clearly measurable. So we here seem to have a system where we can capture most of the ADME protein and hopefully most of the ADME protein activity. We can also see that we have the differences between the donors is often carried on to the organoid level. So the organ donor differences persist in the in vitro system, at least on the rank level. So in conclusion, we say that we can use these enteroids for ADME purposes, at least for intestinal metabolism, and we hope to, in the near future, be able to show also how to use these for transport and permeability studies to make a complement to the CACO2 cells and primary enterocytes that we are using at the moment. And with that, I would like to thank our co-workers, or collaborators in RiskHunter and Signatop, and our collaborators in Mikael Selin's group at the Department of Medical Biochemistry and Microbiology, who really are the experts in in enteroid formation and has taught us a tremendous amount about these model systems. So with that, I thank you for your attention. The image here is a section of human genome that's been exposed to green nanoparticles in an Assing chamber, are clearly absorbing these into the brush border membrane. Thank you. I will be happy to take any questions.
Thank you, Patrick. Do we have any questions in the room? Anyone? Okay. Yeah, go ahead, Steve.
Hi, Patrick. This is Steve Ferguson. Excellent presentation.
Thank you, Steve.
Do you think that there is a concern that proliferation is sort of diminishing the ability to differentiate, or do you feel like the metabolic proficiency that you're seeing is close enough? It looked to me like the metabolism was quite robust, so I was just curious to get your sense of, you know, are we going to need primary cells, or maybe these stem cell type approaches can do the job?
I think the stem cell approaches can do the job, or at least it seems like that now. We have we have two phases here. We have a proliferation phase and then switching to a differentiation media. And that, of course, slows down the growth somewhat. The collonoids are much more robust in shape than the enteroids do. The enteroids do grow more after differentiation. But we can see that the expression of CYP3A4, for example, is approximately a third in the enteroids as compared to the isolated primary enterocytes. And that is also approximately the level of activity we see. So we seem to have a good correlation between expression levels and activity in these different systems.
And we'll be very excited to see your data with PGP and BCRP.
Yes, so will we. And that is very good. What we're also trying to establish is, say, out of 100 enteroids, how many of these are actually tight, or how many of these would take up a paracellular probe like Lucifer or Dexter? And that is something we're working on now, and it seems promising so far.
Good, thank you.
Great, thank you very much. Okay, so we'll move on now to our next presenter. So Dr. Patil is ill and so is not able to present. And we are extremely fortunate to have Dr. Nelson agree at the last minute to step in and give us a presentation on accelerating living microbial therapeutic development with gut-on-a-chip technology. So Tyler, over to you. We see your slides, looks very good.
Are you able to hear me all right?
Yes, you sound great.
Right, I'm trying to figure out how to get the video on, but I'll figure that out in a second. I appreciate the opportunity to talk today. This is my disclosure. Today I'm going to talk a little bit about, there we go, thank you. Today I'm going to talk a little bit about our group's work at the Air Force Research Laboratory. We develop primarily microvascular endothelial cell and gut-on-a-chip models that allow us to conduct these types of both absorption, transport, and microbiome type studies. So first I'm going to go over a project that we did in collaboration with a company called SynLogic. And then we're going to talk about some of the work that we've done in synthetic biology and developing sense and respond microbial strains and how we've tested them in a gut-on-a-chip. Talk a little bit about some future directions and provide some wrap up. So just as some context, at the Air Force Research Lab, we sit within the systems biology CRA, which is a core research area within the 711th Human Performance Wing. Our goal is to ideally try to look at the pilot or warfighter or airman, in this case, all the way from individual molecular kind of background all the way up to human systems level background and analysis, and then develop ways to create predictive models as well as countermeasures to both assess their their performance within different warfighter scenarios, but also develop countermeasures to prevent declination in performance. So for our group, myself and Dr. Matthew Grogg on the call here lead this in vitro team, and the way that we address this is typically using a model development and workflow as described here on the screen. So we work very closely with multiple different partners, including partners there with Dr. Harton's lab, and as well as at Cincinnati Children's Hospital just down the road from us, and at Cedars-Sinai. We work across the board using induced pluripotent stem cells, gut enteroid stem cell models, primary cell models, as well as traditional like KCO2 and other cell line type systems. We have multiple different types of microfluidic-based organ-on-chip systems that are commercially provided, as well as in-house models that we develop. We do a fair amount of different types of data acquisition and try to figure out ways to integrate them into our workflows, utilizing typically very small volumes of affluent or conditioned media to assess outcomes in our model systems. And then probably most importantly for our Air Force relevance, we try to emulate different operational environments as closely as possible, and then use these types of systems to make predictions, test countermeasures, and evaluate molecular mechanisms. So first I'm going to talk about a clinical symbiotic that was developed by SynLogic. This was an opportunity that they reached out to us to kind of see how we might be able to use a gut-on-a-chip to characterize how their symbiotic that they developed for PKU disease to be effective in vitro settings. So could we provide unique data that would allow us to show that there's high efficacy for a given strain? So there's a lot of re-engineering that occurs in developing those engineered strains as they do. And so they wanted to be able to use a more physiologically relevant system to be able to test and evaluate the function of those strains in a dynamic environment. And so we utilized the Emulate Bio microfluidic device. So as you can see on the top left there, it's a 2-compartment PDMS-based microfluidic device that has 2 separate compartments. If anyone saw the talks yesterday, you saw a really nice talk from Emulate about this platform. So it does have a porous membrane. The pores are quite a bit larger. They're around 7 microns and they're hexagonal. These 2 compartments are not exactly the same size. The top compartment is a little taller, about 1 micron in height, and the bottom compartment is 0.2 microns in height. So you get this vascular lumen that's a little smaller in terms of the Z dimension, which allows for a little higher shear stress. We then functionalize both of these different compartments using specified extracellular matrix proteins to allow for the unique attachment of vascular cells versus epithelial or gut cells in the top compartment. We then mature these models within a pneumatic pumping system as seen on the far right. For our gut-on-chip system, we typically mature these for out to 10 days. If we're using KCO2 cells, and if we're using intestinal enteroids, we would go about 8 days for maturation on the chip system. So here I'm presenting a KCO2 HT29 MTX model interfaced with gut microvascular endothelial cells. In the top compartment, we mix the KCO2 cells with the HT29 cells at a 3-to-1 ratio. We then mature these under flow and cyclical stretch, which emulates peristaltic-like forces over about a 10-day period. Utilizing confocal microscopy, we've been able to show that we can fully endothelialize the bottom compartment, kind of creating this blood vessel tube. You see the interaction between the top gut compartment and the endothelium. So in many cases, you can see either green or pink projections that pass through the pores. So you can see there's this nice interaction between the 2 compartments. And then if you look at it on a cross-sectional view, as seen in panel C at the bottom left, you can see that the gut compartment actually is not flat. So what we see over about a 10-day period when we include stretch and flow is a spontaneous morphogenesis that's been explained in the literature to produce these 3-dimensional macrovillus-like structures. And looking at the video on the right, which is a stitching of, I'm not sure if it's plain or not. yeah, it doesn't look like it's playing, but what we would typically be able to see in the video is as it turns and rotates on its side, you can see that these villus-like extensions are actually quite tall. They're around 150 to 200 microns tall, and they're pretty homogeneous across the surface of our microfluidic device. And these projections are important as they provide increased surface area and interaction for microbes to exist within this dynamic flow environment. Otherwise, the microbes would, for the most part, just be passing through the chip and not have much interaction with the gut epithelial surface. So for the study design, Synlogic had already published a couple different studies, both in non-human primates as well as a healthy volunteer study looking at their strain. And so they took E. coli Nissl and they engineered it to sense phenylalanine and then convert phenylalanine into a non-toxic biomarker that they could trace in tranexamic acid. And so throughout this process, they were measuring both tranexamic acid and phenylalanine in the urine and in the blood. And so they came to us and was asking whether or not we would be able to kind of recapitulate some of that study utilizing a human gut on a chip setup. And so on the left is the design or the model of the system that they tested in non-human primates. And in this case, they used E. coli Nissl as their chassis strain. They then engineered it to convert or allow phenylalanine to be uptaken within the strain and be converted into tranexamic acid. And the right is a representation of what our model would look like, where we dosed with phenylalanine and we had different concentrations of the therapeutic strain passing into the chip. We then were able to sample from both the gut effluent as well as the blood compartment effluent and be able to look at physiological concentrations and flow rates within our model system. So for the gut compartment, we utilized a simulated intestinal fluid that included many of the enzymes, a pH that was representative of the small intestine, as well as many of the different components associated with published simulated intestinal fluid formulations. In the bottom, we used a mixture of CaCO2 media with vascular endothelial media to sustain both cell types. And this study was done all at normal oxygen tensions within an incubator. So we were able to publish a study showing that we were able to predict pretty closely how phenylalanine was able to be utilized by the live bacterium and transported throughout the gut. And throughout this study, one of the main features that they really wanted us to make sure that we were able to accomplish was looking at the clearance of E. coli Nissl strain in our model system. So they had engineered the strain to not be able to proliferate, which allowed it to have a unique clearance that was able to be tracked in both the human volunteers, non-human primates, as well as in our model system. So we tuned the flow rate within our model system to achieve about between a 6 to 9 hour clearance rate, which was found to be within reason because they found that clinically a single dose was cleared within about 8 hours. Utilizing a low, medium, high dosing scheme, you can see there's a difference in how long the species are sticking around within our gut system and how long it takes for them to clear. More importantly, if you look at panel D, the amount of phenylalanine that was reduced within the gut was substantial at the high dose. And you can see in a dose-dependent manner, no significant change for the low or medium doses. And then looking at the corresponding production of the biomarker within the gut effluent, you can see a low, medium, high production of TCA that we were able to track using liquid chromatography mass spectrometry. And you can see that the biomarker is much easier to track than phenylalanine, and that's partly because this is an easier phenomenon to capture within our model system, as phenylalanine is already included in our cell culture media. So we were dosing at higher levels of phenylalanine, representative PKU in this particular study, but the corresponding concentrations of TCA were able to be traced quite nicely. And you can see as the therapeutic clears out of the gut chip, you see the reduction in TCA, the reappearance of phenylalanine back to normal concentrations in a dose-dependent manner. We were also able to look in the blood compartment and see the transport of tranexamic acid, the biomarker produced in the gut. And we were able to trace this in a low, medium, high concentration as compared to the no treatment. And most importantly, we were able to show that the therapeutic was able to reduce the circulating concentrations of blood phenylalanine as compared to the no treatment condition. This was all while maintaining a normal apparent permeability based on our calculations using a cascade blue indicator. One major advantage that we wanted to point out was that we were able to do supraphysiological dosing. So one of the things that they had limitations in with the animals or within healthy volunteers was the ability to go to relatively high doses and do multiple doses over a given time period. This was both something that they were interested in and something that they were wanting to be able to see the dynamics of the model system. And so we were basically reapplying a dose of the therapeutic after about 7 hours. And then we were able to track and show that we did not allow it to clear out the gut chip as a function of time. Most importantly, if you look at the pictures on the right, the panel, one of the features that we were able to show was that using DIC and zonal occludum 1 staining, we could show that under a low, medium, high dose scheme where we did not allow it to clear, we saw a significant loss of zonal occludum 1. And we were able to look at the villus height. And you can see that there's a significant reduction in villus height as a function of increasing dose and prevention of the clearance of that strain. And so you can see that you get this villus blunting effect that occurs as a function of dosage. And more importantly, you can see that in the apparent permeability, the highest dose when we don't allow it to clear, which in this case was 10 to the 9th CFUs per mil, you can see that you see an increase in the apparent permeability that was statistically significant against the low or the medium dose. The villus height also became statistically relevant in terms of measuring the actual height of those villus as a function of treatment. And so the nice aspect of this study was to be able to show and predict that supraphysiological doses of this particular strain over this time period and flow rate did have adverse outcome effects on the host model that were also emulated at high doses in healthy volunteers. But I think the major take-home message was we wanted to be able to look to see if the treatment or the therapeutic was actually able to recapitulate how it could reduce circulating Phe levels that would be independent of dosing in the gut compartment, but rather the circulation or entero-recirculation of phenylalanine that might already be present in the body. They weren't able to do this study in non-human primates or in healthy volunteers. So this was actually new data that was kind of enabled them to start thinking about predictions of how it could be used for reducing circulating levels throughout the body. And so we applied phenylalanine in the blood compartment, we applied our therapeutic strain in the gut compartment, and then we modeled or sampled both compartments over time, and we're able to show that using the clinical strain dosing scheme, we were able to maintain villus height at a normal level. We're also able to maintain the apparent permeability, but most importantly, the area under the curve for phenylalanine in the blood compartment, which was continuously being dosed, was actually reduced at the highest concentration as relative to the lower, medium, or no treatment conditions, suggesting that a therapeutic strain applied just to the gut alone was able to reduce circulating levels of phenylalanine in our model system. This was great, but I think the major outcome that they really wanted to make sure was how predictive was this model and how useful could it be for future engineering efforts of ecolanisol or other therapeutics in terms of down selecting high potential strains for non-human primate or even healthy volunteer studies. So they took the PBPK model that we developed for the non-human primates, and then we also constructed a PBPK model for our in vitro system that looked at the transport of phenylalanine and TCA as a function of the apparent permeability calculations that we made with our FITC-dextran and cascade blue indicators. And then we used that to then characterize the level of correlation between our data and what was produced in the non-human primate. We were able to achieve a Pearson's correlation coefficient of 0.72. And this was very important in terms of their understanding of our model system and both how they could potentially utilize this model system in the future. And for us, it was a great validation to show that this system provided a good platform to characterize transport and barrier integrity studies moving forward. Now, switching gears, we had a internal call to develop synthetic biology capabilities through an ARAP. And through this program, one of the goals was to find ways to utilize the gut and the microbiome and probiotics, in this case, synthetically engineered probiotics, to potentially augment the gut-brain axis by eliciting a neurotransmitter. So in this particular system, we wanted to be able to look at both the impact of like disease models where you might have stress or neuroinflammation that's impacting cognitive function, and be able to utilize the gut as a way to potentially mitigate or make our warfighters a little bit more resilient to these stressful environments. So utilizing a similar gut model system, still using Keiko2s, we wanted to look at a engineered symbiotic, in this case, still using E. coli Nissl, but in this case, it was able to sense cortisol as its stress molecule. And upon sensing cortisol, turned on a genetic circuit that converted bioavailable tryptophan into tryptamine. We then traced cortisol, tryptophan, tryptamine in both the gut and blood compartments. We quantified how much of the E. coli Nissl was actually growing as a function of time. So that's a major difference between this study and the previous studies, that this particular engineered strain is actually proliferating in real time. And so we actually had to dose at a much lower concentration and allow it to grow within the chip over a given time period. We then applied physiological levels of cortisol and L-tryptophan to monitor tryptamine production. We also had an engineered strain that had a GFP tag in it because we wanted to understand how E. coli Nissl was distributing throughout our chip as a function of time. Hopefully this video plays. So as the video steps through here, you can see the green glowing E. coli Nissl are kind of sitting in between these villus-like projections. This was a good outcome for us to be able to show that we actually were able to get some E. coli Nissl that stayed within the actual chip and wasn't just floating through the lumen. And in this case, we were able to show that upon cortisol stimulation, that it actually glowed green. And we were able to show that that biosensor was actually working within this gut model system. So we then wanted to characterize the viability of our model system as a kind of a function of time and cortisol exposure. So one of the interesting aspects was that if we went to super high levels of cortisol, it actually impacted the viability of the model quite substantially. So we had to stay within that physiological range, which is still quite high, but it's within physiological realms of 1 to 5 micromolar concentrations of cortisol addition. We then monitored the wild type E. coli Nissl strain and the synthetically engineered SYN strain with and without cortisol activation. You can see that with the synthetically engineered strain, we had slightly different microbial growth characteristics as compared to the wild type or the synthetically engineered strain without cortisol. And this is likely due to the activation of that genetic circuit, reducing its ability to proliferate. We then looked at the apparent permeability as a function of a given input of E. coli Nissl over time. And so we inputted a 10 to the 7th CFUs per mil, and then it grew to about 10 to the 8th or 10 to the 9th CFUs per mil within a 24-hour period. You can see that the E. coli Nissl and the synthetically engineered strain did increase the apparent permeability, but not substantially without reason to be within a range for typical human apparent permeability constant conditions. We then did it with and without cortisol, and you saw a little bit more deviation for the synthetically engineered strain as compared to the E. coli Nissl wild type strain. This outcome was somewhat surprising because the only thing we did was add cortisol, and we assumed that would impact the strains all the same, but we actually had a slightly increased amount of apparent permeability change as a function of cortisol addition at both 12 and 24 hours for the synthetically engineered strain as compared to the wild type. So then we wanted to trace tryptamine and tryptophan production both in our gut and blood compartments. And so in this case, metabolism is actually metabolism by the E. coli Nissl strain. And not we didn't monitor it within the gut cells, and although the gut cells are also metabolizing it as a function of time. So what we did is we monitored additions of normal physiological levels of tryptophan in the gut, and then we characterized it with or without the wild type or synthetically engineered strain. And as you can see, at about 12 hours, we had pretty substantial and very quick metabolism of the tryptophan that was available in the presence of the cortisol stimulant. In this case, that was a 5 micromolar stimulation of cortisol. And then correspondingly, you saw an increase in tryptamine that both increased substantially at 12 hours and then spiked at 24 hours relative to the wild type or no treatment condition. Importantly, you see this kind of delayed transport of the tryptamine within the blood compartment, so it happens post 12 hours. And you also see that it corresponds nicely with the amount of tryptophan that's being transported across that barrier as a function of time. Interestingly, at 6 hours, you can see that there's very little cytokine activity that was impacted by both the wild type or synthetically engineered strains. They are statistically significant, but we're looking at no more than a 2-fold change here within this heat map as compared to the no treatment condition. However, at 12 hours, the synthetically engineered strain really increased substantially in terms of its cytokine activity. And this is likely due to it completely depleted all the bioavailable tryptophan in the gut compartment and most of it in the blood compartment as well. This outcome was actually a really good feedback to our engineering team that was developing the E. coli nasal strains, as we needed to have some kind of feedback mechanism that would potentially stop the production of tryptamine once tryptophan was depleted or close to being depleted. So we also wanted to test the quality of the engineered strain within the model system. So how much cortisol was needed, and if we didn't have full induction of the genetic circuit by the cortisol, did we still produce tryptamine in a meaningful manner? And so we came back and did different concentrations of cortisol within the gut compartment. We then monitored how much tryptamine was produced and how quickly the tryptophan was depleted. So you can see at partial activation of the genetic circuit at 1 or 0.2 micromolar cortisol that we don't get full depletion of the tryptophan and the gut compartment, and we don't get full production of tryptamine. So it was a linear response based on the induction of that genetic circuit as a function of time. We also looked at the opposite scenario where we had excess tryptophan or reduced tryptophan as a function of time. And you can see that the tryptophan was actually at 10x was still able to be significantly reduced as compared to the no treatment or the E. coli nasal strain conditions. But at 100x, we had so much excess tryptophan that we could not fully remove all the tryptophan from the model system, and it saturated at around 150 micrograms per milliliter in the tryptamine production in the gut compartment at 24 hours. So we got significantly more production of tryptamine as a function of increased tryptophan, but there seems to be somewhat of a limiting amount of tryptophan that the coli nasal cells were actually able to process. So moving into some different studies that we're doing in our labs and where we're trying to head things, most of the work I've presented thus far, or all of the work I've presented thus far, has all been CACO2 models. So there's obviously a big push to try to use more complex model systems and have better transporters represented in the model system as presented by many of the presenters over the last 2 days. So what we have done here is we've utilized intestinal enteroid-based cells from the duodenum and the colon, and we were able to reproduce intestinal on chip models utilizing the emulate microfluidic devices. And so you can see they produce a pretty robust and unique looking macrovillus-like structure on the surface. We then were able to stain and see nice production of MUC2 and localization of villan within our model system. And really the goal here is to increase the complexity of the model and then be able to use those for unique scenarios that require the full complement of transporters as well as gene expression and metabolism as expressed in this workshop. But another big feature was we wanted to move more into doing full fecal microbiome studies. And one of the big aspects there is to be able to control the oxygen environment within the gut compartment. And so what we've done is we've integrated real-time oxygen sensors within our microfluidic system that allows us to monitor the oxygen content in the effluent as a function of time in both the gut and the lung compartment, or the gut and the endothelial compartment independently. We also built this microfluidic-based system inside of a glove box as pictured there in the bottom left. This allows us to control the atmospheric oxygen environment surrounding our model, but we also have the ability to input different oxygen concentrations into either the gut or the vascular compartment independently. And so on the right, I just wanted to present an image showing how well we were able to control the oxygen environment within our system. And so we developed this oscillation program that allows us to move the oxygen content within a 30-minute period between 7.25 to 11.25% in the bottom compartment, and the lower compartment go from 3 to 0.3 within the same time frame. Very quickly. And so we have pretty good control in both the vascular and the gut compartment in order to control the oxygen environment surrounding the chip. And we utilize this glove box system to enable us to run these full fecal microbiome studies. Of interest to us right now is also connecting these with traditional microbial batch fermentation model systems that would allow us to grow up and conduct different types of interactions of complex microbes with different types of dietary fiber, as well as different dietary inputs. We also can do different exposure scenarios to different chemicals, or look at interactions with potential pathogens. We can do these on chip as well, but the one advantage to doing this in batch fermentation is we can expand it. We can also monitor different regions of the intestinal tract in the batch fermentation, and then pull that effluent sample out, pass it through our model system, our model host system, and characterize these responses over long periods of time. So we would be able to take fecal samples that have been growing and be able to characterize them at different stages within their growth characteristic as a function of the experimental time. And so one example of this is we just recently conducted a large hypothermia study in which we were characterizing host microbiome interactions at temperatures that represented extreme hypothermia versus extreme hyperthermia, with our preliminary results basically indicating that you get significant changes in the production of or sustainment of microbes within those different model systems, both anaerobic and under aerobic conditions. So we're very excited about using this system moving forward to characterize these more complex host microbiome interaction situations. In summary, we've developed some dynamic organ-on-chip models that represent the gut, and we were able to screen and test several different types of symbiotics, kind of suggesting the opportunity to reduce animal needs at those early engineering circulations where you're trying to optimize different strains for different purposes, and really look at the efficacy and function of those strains prior to introducing them into more complex animal systems. We've been able to integrate in silico techniques to really allow us to do predictive characterizations, but it also allows us to optimize the conditions for our gut models prior to doing those types of experiments as well. And our lab also works in other types of organ systems, so the brain, muscle, and lung, for example. We've integrated these types of model systems as kind of a, not in the same platform, but as a kind of manual characterization or pullover of the effluent into different model systems, with the goal of really starting to connect these systems directly and have gut-brain axis, gut liver, as well as other types of model systems within our laboratory to not only test symbiotic function, but also look at the stress impacts of different types of operational environments. I thank you for the opportunity. Have a good day.
Thanks, Tyler. Really fantastic work. I'll open it up for one quick question in the room. Gonzalo?
Yeah, thank you. So very nice presentation. I mean, a lot of data, so I'm still digesting part of it. So I'm curious about the model that you developed for fatty alketonuria. And one thing that's confusing me is that the traffic of amino acids through the gut is polarized. I mean, it's driven towards absorption. So I understand, I mean, the concept of your model, but doesn't it simply reflect the fact that you may not have proper polarity in the permeation of the fatty acid of phenylalanine? I mean,
So
would this be expected to work in a functional human gut?
Yeah, we would expect transport of phenylalanine across the gut, but it's also being metabolized by the epithelial cells within the gut model system as well. So the model is both looking at the natural metabolism of phenylalanine, but also the transport of phenylalanine across that gut-blood barrier.
Okay, all right, okay, thank you.
Okay, great, thanks. In the interest of time, we're going to move on, but please, attendees, continue to put your questions in the Q&A box. And Tyler, if you could address the one that is there in writing at the moment, that would be great. Okay, our final presenter for the morning is Dr. Colin Bishop, who is from the Wake Forest Institute for Regenerative Medicine. We'll be speaking to us about gastrointestinal toxicity in model animal species using organ tissue equivalents.
So I get the pleasure of being the speaker that holds you up before lunch, but I'll do what I can. Oops, is it this one? Yes, hi. So thank you all for inviting me. Our lab is not 100% invested in the gut, but nevertheless, I've got data here that I hope you'll find interesting. Okay, the project I'm going to talk a little bit about is our DITRA-funded Noah's Ark project. And the problem that DITRA came to us for was, of course, animal models, as you all know, are not terribly predictive of drug outcomes in human. And the problem was they wanted us to put together a system where they could perhaps identify the best animal model to use for both MCM approvals and at the time there's an animal rule which the DOD had in place, which I believe is a little bit less strict now. Our solution was to construct functional organoid equivalents, tissue equivalents from multiple species and organs and compare their responses to the drugs with our human-derived OTEs. The procedure was after the characterization of the OTEs and they're screened with known drugs, and the biological responses were recorded. We could evaluate the differences and suggest which animal would be appropriate for their particular drugs that they were interested in. And also we were hoping that long-term, this sort of technology could help to replace the use of animals in drug development. The species that we chose to look at were human, mouse, rat, dog, and primate, which are often used in experimental procedures in the lab. And we chose 3 organs to look at: the liver, lung, and the gut. We decided not to use cell lines in any way. All of these are derived from primary cells. We mixed the cells together in approximately the same proportions that you would find in a human liver, for instance. So just for instance, in the liver, I think we use about 75% hepatocytes, I think now about 10% Kupfers, 5% stellites, and another 5% of cholangiocytes. All of these we source from commercial sources. We also thought, okay, how are we going to do this? In our lab, we have a lot of different organoids and a lot of different ways of making them. For instance, we have planar lung organoids, which we're using for another very large DOD project, which again contain all of those cell types, but in a planar fashion. We also have guts, which contain those cell types, again in a planar fashion on trans wells, of course, as has been described. But we decided, no, this is too tedious to make them on these trans wells, and they're not as high throughput as we want. And we would like to be able to eventually use high throughput multi-image screening. So we decided to make everything as spheroids. And simply, this is how one does it. It's extremely simple. The cells are taken, viability checked, mixed together, put into either a 96 or what we're using now is 384 well plates. They're allowed to sit for maybe 96 hours till they compact nicely, and they remain viable for about a month. For instance, here we have the liver, just shows you that it's very reflective of a human liver. You can see its functionality over about a month, and pretty much the same with the mouse and the other organoids too. You can see we have Kupffer cells in there, the stellates and so on and so on. And we also can see where we do an EM that you can see the caniculus cells. Little, caniculus is developing there. We also have looked at the transporters and the CYP enzymes, and also metabolism over a month. And it all, this is our sort of quality control. It took us rather a long time to identify the exact cells. Many of the commercial sources of these cells are not exactly what they're meant to be. So it took us some time to get all this worked out, and the media and so on and so on. Again, with the rat, mouse, human, rat and mouse and lung OTEs, you can just see this is just an overall quick look to show you that they are fairly representative of a lung, but in the shape of a spheroid, and all the cell types are in there. The lung is also interesting because if we leave it for about 12 days, you can see on the outside in this film all the little villi, moving. And we actually use this in another project, the movement of the villi as a functional output of the drug screening and so on. Finally, of more interest, of course, to this audience, we've taken the small intestine gut OTEs, and they've just been formed in the standard way according to Hans Cleaver's. They are all derived from the duodenum. So we have human, mouse, rat, dog, baboon. And we, again, we played around with them a little bit so that we could get them all growing nicely in a single media. Commercially, you can buy media for human and mouse and so on and so on. And it's rather expensive. We have pretty much made our own media, and all of the cell types seem quite happy, and we can mature them nicely. This is just simple characterization of them, which I'm sure you've all seen from the previous speakers. We see very similar things. And then what we have done, at least in our initial screening, we've taken just 6 drugs, some universal toxicants like cisplatin and arsenic, some metabolites, diazepam, propranolol, which we measured their metabolism by mass spec and so on, and some known hepatotoxins like APAP and aflatoxin. And over a range of dilutions, because these are all in 96 well plates, we've measured the IC50 values. All we do is after 7 days after seeding, all of the organoids have compacted. We add the drugs, and then after 3 days, we do a simple assay, simple ATP toxicity assay to check for viability. We're hoping that we have a more sophisticated instrument that we should be able to do some high throughput imaging. Overall, of course, what we're looking for, what DITRA is looking for, and what we are hoping to provide is a system which will reflect differences in the species. One of the known differences, certainly in the liver, is aflatoxin B1, and you can see here that the human liver is very, very sensitive to it, whereas mouse is much more resistant, approximately 20 times. And the areas in yellow are areas where we can show that there are differences between these species, and these are known differences if you go back and look at the literature. We have the main thing about this is to try and get a working SOP, so it's reproducible, so we can do it on Monday, we can do it on Wednesday, and we can do it next week, and we can get exactly the same result. And this is what we're working on now. The results I've just shown you are sort of our initial run-throughs, and we're very confident now we can do that. So we've stepped it up actually to 384 well plates. We're moving forward actually to 24 drugs, and we should be able to screen all of those organoids fairly quickly and produce this sort of model system. And just to change gears just a little bit, I am also collaborating with a colleague of mine, Dr. Steve Walker in our lab, who's very interested in autism. And as you may know, many autistic children have gut problems. And so we were interested in making a sort of a biobank of gut organoids from autistic children. So we obtained small biopsies of 1 or 2 millimeters, which were sent to us from collaborators in Texas. And we were able to convert those into gut organoids. We have been able to, isolate, of course, the crypts and establish the steroids. We have them stored down in nitrogen. We have, it's interesting actually, because these are all from children of about 3 years old. And I would have thought that would be difficult, but it turned out to be quite the reverse. These tiny little biopsies grew like crazy. I assume, and I don't know, I assume either they were from, that there was something wrong with the gut itself, which allowed it to proliferate fast, or simply that it's younger tissue. Our longer term objective, if you like, is to link the autistic guts to a brain model. So we have our own in-house integration models, and we want to integrate not only the previous screening into a 3 organoid model, which we actually have, but it's not quite ready for prime time. But we also want to link up the brain and the gut. And one thing we're highly interested in is actually in the enteric nervous system, which is if you like, the other nervous system, which nobody pays a lot of attention to. But overall, we have, from the autistic project, 39 viable samples, 31 from autism spectrum, and 8 viable control samples. It was the controls that were most difficult to get because what surgeon biopsies a normal child, but we were able to get them. And they're stored down. The great thing about these, of course, is with all of the gut organoids, they can be expanded for an awfully long time, and low passage vials can be produced. The inventory in August, it's all a bit strange, but we have them from 3 areas: the duodenum, the right colon, and the terminal ileum. We don't have a choice about what we can take. We just, get whatever the surgeon's willing to offer us. We're slowly building this biobank up, and of course it's going to represent a whole spectrum of autistic diseases and also different genotypes, presumably in the children. So in summary, we've produced functional OTEs from human, mouse, rat, dog, and primate. I didn't show you the dog and the primate because they're all fairly similar. We've shown they function over long terms. The liver, we can very functional up to maybe 30 days. The lung, we can keep going for maybe 6 months. And the gut, of course, you can, you have to keep passaging it, but it can go for an awfully long time. We've screened all the OTEs from the species of the 6 drugs, and I think what we have achieved in our initial work is to standardize this, and we want to now go into a more high throughput screen with more drugs. The other thing we want to do, and we hope we're going to get funded to do it, is that we could make the argument that all this was a waste of time. Because how do you know that if you screen an organoid with a drug and you get such and such a thing, how do you know that that's what's happening in an animal? So one of the things we're doing right now is that we're going to go back to look at and get as much data as we can. We're going to choose maybe 1 or 2 models and see the effects of that drug in vivo from hopefully from published data, or we might even have to do it ourselves, and make sure that the OTEs are accurately reflecting what actually happens in vivo. Otherwise, they're not terribly useful. The final thing, of course, is this is another project. We're developing this biobank of intestinal organoids from autistic children. And I think once we have that linked up with the brain and actually in an immune system, we have a rudimentary immune system, which is tricky because, of course, any immune system is going to react to the organoids you put in because they're not all of the same genotype. But we have a way we think that we can get some sort of reasonable immune system in this context. But I really want to thank the organizers for letting me present this work, and of course DITRA for the funding, without which we wouldn't be able to do any of this. Thank you.
Thanks so a lot, Colin. It's never been called an organoid before. It's a new one. Any questions in the room yet from Soliger?
Yes, that's a really nice presentation. I'm really interested in your model to try to ascertain directly with the effects on given organs. So one thing that I always have in mind is the fact that more often than not, it's not the parent compound, but rather metabolites that are the toxicants. And so in light of that, is it possible for you to put organoids derived from liver in co-culture with the organoids from target tissues that you're interested in to make sure that you have the liver metabolism recapitulated there?
I'm sorry, I didn't quite hear that.
Oh, I'm sorry. Okay, so I always worry that you of trying to ascertain toxicity of a given compound if you don't have metabolism present that can produce the metabolites that are frequently more toxic than the parent compound. So I mean that is sort of self-contained in the liver organoids, but not necessarily in organoids from other tissues. Is it possible, and this is just a practical question, is it possible to do co-cultures of liver organoids with organoids from tissues of interest so that you have metabolism?
Yes, we've actually already done that. We published a 6-organoid system a few years back where we showed with these 6 organoids, if we put in a prodrug and left out the liver, the heart in particular, which was affected by it, would just keep on beating. Mm-hmm. When we put the liver in, you really didn't need an awful lot of livers, and it was highly toxic to most of the organoids, especially the heart. Mm-hmm. It is. What the screening system is just a quick one pass to see if there's any endogenous toxicity in that particular drug. Mm-hmm. I mean, if we find one of those drugs is really killing maybe the liver or anything, just non-specifically, well, then you can throw that away. After that, mm-hmm. You're absolutely right. You need the liver in any system to, virtually all of the cancer drugs are prodrugs, aren't they? Which either get activated through the liver and so on. Mm-hmm. So yeah, you're absolutely right. It's just tricky business to do a high throughput multi-organ system, but we're getting there actually.
Yeah. In a sense, I mean, this simplifies the engineering challenges of having to concatenate multiple compartments. And so, I mean,
well, what we're doing, we have another project in the lab where we're screening lung organoids. These are planar organoids on trans wells, and we're looking mainly at a whole host of viruses, but we're also using them for a limited number of drugs. So we make them, just as has been described before for the gut, but it's actually the same, with the endothelial cells there. But underneath we have a 96-well transwell plate, but underneath we put 2 or 3 liver organoids. Mm-hmm. And so that's just a lung and a liver, but it's very, very easy to do because you make it in the same plate. You put the liver cells in, and then you put the transwell, which is one big transwell, which you just pop in. So that's one way we're doing it, but we have more sophisticated ways. But this was started a few years back, and it was just a quick screen. But we really want to make sure that those results actually reflect what's happening. What ultimately we would like to do, not necessarily for DITRA, but to compare a clinical trial in vivo, a real one, the clinical trial on a chip, if you like. But you know, getting a drug company to agree to that is almost impossible.
Yeah, that would be a really big chip. But thanks a lot. That was really interesting work. Thank you.
Great. Thank you so much. Okay, actually, Colin, you are not the last in between us and lunch because if you'll recall yesterday, we went pretty late yesterday afternoon and so decided not to do the report outs from the breakout groups yesterday at the end of the day, and instead we're doing them now before we break for lunch. And so I asked Dave and Lynn to try and keep to about 10 minutes each and hit sort of the salient points because we're also very fortunate, thanks to the support of Thomas's group and Kat from Johns Hopkins, to have a science writer, Susan, here with us, who's been taking diligent notes and is going to be following up with a draft of a workshop report. So yeah, Dave, I'll turn it over to you for the first report out from Breakout Group A.
Great, thanks, Nicole. So I'm just going to go through the establishing confidence group. Nicole and Susie certainly kept everyone on task and worked through the questions while myself and Kemi frenetically moved through taking notes. So here are the questions, as you'll recall, that we tackled collectively. And I'm going to just take a few minutes to summarize 3 hours worth of discussion. So the first, what are the most critical needs in each of the areas covered? And I think a common theme when we get to that last one, evaluating potential allergenicity, I think we were a little ambitious in including that in this workshop topic. So we have limited responses, if any, there. So we'll focus most of the discussion on frank GI toxicity, de-risking systemic tox, and understanding absorption and pharmacokinetics. So certainly there was recognition that most of the initial efforts in this field have arisen from the pharmaceutical industry and some simple de-risking scenarios to move compounds forward or not, and the questions being asked there. But certainly as we move into more complex matrices, obviously that matrix matters, and the examples that were commonly talked about were like food matrices and complex mixtures that we are exposed to. So obviously needing a model that can model realistic exposure scenarios, again, the matrices, clearly something that has digestion and a metabolic component to it, so that what are we actually being exposed to can be defined. And certainly that common theme of having a metabolically competent method in place. Also the concept of gastric residence time and certainly physiologically how important that is, how long something resides in the GI is important to that question. And one example, no. Okay, great. Sorry about that. I believe it was Gonzalo that talked about an example of hepatocellular toxicity associated with lectins that induced some hepatocellular hypertrophy, I believe, but in the absence of digestive processes, and how relevant is that? We also talked in the absorption realm of certainly there should be some sort of simple Franz cell system akin to those used for dermal absorption, and how that could perhaps be put in place. Again, the concept of biotransformation and metabolism being the more complex issue there. Emphasis, I believe it was Dr. Sayers that highlighted the importance of histological evaluations to fully understand and characterize model systems. And I think we've had some wonderful examples this morning of microscopy and the power of imaging and fully understanding and characterizing these model systems. And of course, the microbiome, what role it plays, how do we take into account geographical differences? Certainly that's a problem throughout the field. Certainly emulates using some synthetic biology, But finding media and cells that can thrive in the presence of bacteria is certainly not a trivial task. And certainly, along those lines, what are the changes to the microbiome associated with toxicant exposures? We also talked at length, really, about the difference between technical and biological variability, and are we seeing variability in the system associated with conducting the assay itself, or is it actual inherent biological variability associated with the cell system? One idea that was brought forward was, having a comprehensive atlas of the entire gut, really, from a transcriptomic profile, cell type, mucus layer, microbiome, you name it, having a comprehensive reference point. And certainly, I think Dr. Wambau brought forward the Certara and Simulations Plus that have successfully broken down the gut into 20 different compartments and models associated with those. But certainly, we need to avoid the over complexity of model systems to be able to answer the questions that are being asked. I can't remember if it was Susie or Paul that mentioned the FDA's list that they have of gaps or areas where existing methodology could perhaps be improved. So, certainly reflecting on that list as we think about GI toxicity models. And, really probably should have started with this, but an overarching theme along really all of the questions is defining that context of use. And, what are those regulatory needs and questions being asked? And I think it was Elijah Peterson that mentioned we don't need a laundry list of research topics. We need to address specific questions based on context of use and regulatory needs. Any questions or comments before I move on to the next question? What areas would you consider most ready for application? For GI toxicity, overt toxicity associated with acute exposures. I think, those are in place today. NAMs are available. As this was actually yesterday that that comment was made, but it certainly has evidence by PowerPoints presented during day one presentations. Barrier disruption, considerations of mature versus proliferative cells, can the gut heal itself, are also important considerations. But not yet, we don't have models in place yet that can recapitulate remodeling or other long-term effects at lower levels that are certainly important when we think about GI toxicity. KCO2 models, obviously, as we've seen throughout the program, are routinely used, but depending on the question that's being asked, whether or not they're actually ready or implementable at this point. And, in some cases, the example is given, perhaps you need a more defined model. If a specific transporter is needed that's not represented in KCO2 cells. They certainly can address cytotoxicity effectively. Dr. Ferguson talked about the high throughput nature of using TIR in those assays. Certainly the permeability assay is very useful, but a lower throughput. We need NAM. We have NAMs with dynamic systems, some complex continuous flow incorporated in them to model interactions at steady state. We've seen that organoids are certainly tunable to different cell populations by modifying culture conditions, that we can explore mechanisms of toxicity and looking at things like pathway activation. Another common theme that we've talked about this morning again was recapitulating human diversity and the critical importance of that as we think about regulatory questions. I think it's fair to say, certainly in this space, that the NAMs are already better than the current models or methods that we have in place for evaluating gastrointestinal toxicity, because we have folks like those at Alt Systems that are working towards building biobanks of diverse populations. And certainly, again, models to characterize metabolic pathway perturbations are still needed. Any comments or questions there? Okay, examples of successful outcomes, and the original question was, what are examples of less successful? We actually modified that to say in progress applications of NAM. So again, successful overt toxicity from acute exposure. I think there was universal agreement there. Permeability, intestinal permeability. Although one comment that was made that there are certain biases that exist in using intestinal permeability models, and perhaps more blinded studies are needed to fully establish confidence there. Metrology approaches, particularly things like LCM mass spec for compound identity and recovery are quite successful. A couple of pharmaceutical industry examples where patient-derived organoids have actually been used by Vertex to expand their indication of the cystic fibrosis drug, as well as Dr. Nelson today talked about synthetic microbes for the treatment of PKU. Recognition that small molecular weight compounds need different considerations than particles do, like nanomaterial. Multi-species organoids to evaluate species-specific toxicity. We just saw a really nice example of that from Colin. And of course, several in silico successes that Dr. Wambau kicked off the agenda today with, certainly in the ADME space, like SimSip. Again, the Certara and Simulations Plus compartmentalized models. Very effective for screening and prioritization, de-risking, recognizing that maybe you might exclude promising compounds, but certainly they can also be used to fill data gaps. And another example, the collaborative acute toxicity model suite for acute oral toxicity and lethality is already a success in place. And a couple of in progress, again, recapitulating long-term toxicity, developing models there, things for the inflammatory or inflammatory cascade or immune response. And then one thing we talked about was about a decade ago now, I think Dr. Wambau published toxikinetic triage approach that could certainly be implemented in this space. Any comments or questions there? Okay, criteria for model maturation, again, defining that context of use and what, we first need to define the context of use, then what do we mean by maturation and what are those criteria associated with that? And what's the consequence of a wrong answer? If we use a method and come up with the wrong answer, depending on the context of use, the severity of the consequence could be quite different.
Hey,
on
1,
the-
2, hey. Okay.
Thank you. Thanks. Okay,
So on the
Gonzalo.
commercial availability, I mean, I think at least part of the discussion pertained to continued commercial availability. Not so much whether people can buy it at this stage, but whether the solution, upon being tried and accepted, will still be available 1 year from now, 2 years from now. And this is obviously associated with the high speed at which things are developing. Thanks. But now that we're getting to the end, I mean, I also, to reinforce what Susie just did, I mean, you did a really good job on summarizing and herding all the cats.
Okay, Len, you want to go next? Yeah, I'm sorry, Paul.
This occurred to me only slowly. I think it might be maybe question 3. Yeah, I think we gotta, you gotta double check that example from pharma, because I don't think they were patient-derived organoids. I think I was just looking at the label. And it was like rat thyroid cells that were transfected with the CFTR. So
I got to be honest, I don't know who mentioned that yesterday.
Yeah. So anyway, double check that one. And then I have one really quick question, which is probably silly, but I think it's the second question. What do we mean by geographical? Where is it? Is that there?
Yeah.
It was the microbiome. Do we mean like, you know, you guys from North Carolina, since you're eating barbecue, and I'm from Maryland, and I got Maryland crabs. I mean, that does make an impact. So
that's what I was assuming we were talking about, not necessarily geography of the gastrointestinal tract.
Right, that's what I was questioning. Right. Okay.
Right.
Or dietary. Yeah.
Our barbecue is better, by the way. Yeah.
It's a different debate.
Okay. Are you going to stay there, Len, or you come up here? It's up to you. Yeah. Sorry.
Okay. Screen share isn't working.
Nathan, can you promote Lynn to a panelist, please, so that she can share her screen? Lynn Crosby.
Stand by.
Been promoted.
They are rejoining now.
Great, thank you so much.
Okay, so I get the dubious honor of being the last person to stand between you and your lunch. So for strengths and limitations of the different model systems, we talked about that regulators do not value NAMS data as much as rodent data, and why that is not understood. This is an area where we do not have much data. Rodents are indeed poor at predicting gastrointestinal toxicity, and we don't have kinetic data for absorption for industrial data. We therefore have a chance to establish NAMs before the animal model is established. We discussed a number of challenges in the first session. For instance, in the last 60 years, we relied on guideline studies and uncertainty factors. Now we're contemplating the idea of moving to new paradigms that are not yet appropriately characterized. Guideline assays took a long time to develop, and NAMs are still in their beginnings. We don't know what the statistical N is. If we have, say, 10 MPS chips, is that an N of 10 or is it an N of 1? Decision-making has relied on applying statistical interpretation to the data, and we've not yet seen a discussion on how to use NAMs in the context of statistics. Most presentations included a control. A lot of the data from the NAMs is sparse with regard to confidence. The responses don't always include a negative control or positive control, might be overtly toxic. And then on the mechanistic side, how do human cells respond to toxicants? NAMs can help us understand that, but we don't have appropriate dose response. We don't have appropriate statistical results, etc. For the gut, we don't have an animal model, so it's not relevant to our discussion. Again, we said that we could go ahead and be working on that in NAMs since we don't have that yet. We don't have a clear understanding of the power we need to make a decision for a guideline study. We're seeing reproducibility and repeatability, but we don't know how to make that power calculation. Calibration, NAMs need to be calibrated. Potencies and efficacies need to be bracketed so your NAM is calibrated, and then as a risk assessor would understand it. Chemicals in the same context would then be evaluated in the same manner. And in the last discussion, metabolism came up as a big gap. And then again, we returned to the idea of the positive control materials for the model we developed. The model is good for some things and limited for others. We are trying to assess barrier integrity, antigen transport, metabolism. For those endpoints within the model we have for PFOA, for instance, this seems to be a good positive control for barrier integrity. Bisphenol A affects antigen transport. Glyphosate could be a good model for antigen transport, but there are no animal models, and so that's why we can't validate them for those endpoints. We agree that the positive and negative controls are needed, but we can go deeper. For instance, if we say the model is good for X, we can find the right controls for X, but they might not be the right controls for Z. We're moving closer based on other peer-reviewed literature. Allergenicity is something we have guideline tests for skin, but not respiratory, but no relevant effort to develop a gut sensitization model. The human problems are complex mixtures of botanicals and natural products. So first, we need examples which can serve as reference compounds, but we can't define them. The comment was made that these are coming from nutrition journals where they could find positive and negative controls for gut irritation versus sensitization. GI side effects are the reason for failure of drugs commonly in clinical trials. It's difficult to predict in rodent models, probably because the GI tract is so different. Ultimately, it's human data that is the gold standard. A lot of the data is proprietary. However, drugs that have already been on the market could be used as examples against which to benchmark. And it depends on what it's for, such as bioavailability. But those comments were not specific to the gastrointestinal tract. They were not specific. GI toxicity makes better sense for the endpoint, but for sensitization, the advantage of the animal model is you can measure the downstream toxicity. Something is not taken up in the gut, then obviously you don't need to do systemic tox studies, for instance. Endpoints and exposures need to be considered. Surrogate measures, for instance, cytotox or change in barrier function. Clinical and phenotypic measures need to be related to cell-based measures. We should continue to collect gene expression, such as Luminex data and Luminex data, but the data is hard to make sense of. If they're going to be biomarkers of a phenotype, we need to collect the data now, even though we can't yet use it. Diseases of the lung are connected to biomarkers expressed acutely. There's a challenge of regulating on non-apical endpoints, surrogates, for instance, loss of tight junctions or loss of water and diarrhea. We should seek human data sets, and we should seek data sets with diversity, such as males, females, ethnicity, and race. Fecal markers, for instance, the presence of blood, these could be conflated with other markers. It would be helpful if there was an established biomarker, such as a change in the microbiome population. It would be great in the future to bring in microbiome people to discuss these issues. The lack of animal models should not preclude us from moving forward with these issues. Animal models are not a good predictor of bioavailability, for instance. This could be an opportunity because, as previously stated, there's no such model. We need to look for the human data and characterize substances just in these models to see what translates to humans. Develop biomarkers out of the NAM model. As soon as models are immunocompetent, it would be interesting to see the substances that differ between skin and airways, and if they tell something meaningful here. NAMs could help to do PK out of the same model in understanding drug handling effects. And we don't push ourselves in these other areas because we do have animal models, but here there's justification for pushing the gut model because we do not have such. Measuring drug concentrations in an immunocompetent tissue model can help establish efficacy and minimum effective dose. The majority of studies seen online with food allergens require sterile regular mice and to look at what shifts in the microbiome or what probiotic affects that. That's where it is heading, but would require funding. Microdosing in humans to get ideas about uptake in humans, or use natural experiments, for instance, food additives, to find how much is absorbed would be a good area to focus on. Isotope labeled bisphenol A were used with very low doses, and microdosing approaches are meaningful, especially with complex matrices like food. In the food safety space, the co-absorption of lipids and so on is a challenge. There's a challenge in adverse events, and we need to rely on what is available in the literature. The second group, again,
Lynn, sorry, just to jump in. This is amazing amount of detail, but maybe save you from reading 12 pages to us verbatim. Maybe do we have like a summary of high points? I don't know, Steve, you were the other person in there. Is there something that distills down sort of the takeaways from breakout group discussion?
Yeah, I actually emailed those and just shut them down in my computer.
Oh no, I should have asked.
I think the primary takeaways were that, you know, for those three different emphasis areas, that there's some key gaps. If you look at that first question, and metabolism and barrier function seem to be sort of the most consensus among the group. Most of our conversation centered around the de-risking side and the pharmacokinetic, toxicokinetic side of things. And what we're trying to do essentially is sort of marry different technologies in a fit-for-purpose manner. And so a lot of conversation around that. There was some conversation about the GI toxicity and different questions we might answer. I think that the questions people are asking are more nuanced from a regulatory sense, but I think there were good suggestions that were documented in there. There was very little conversation about allergenicity. We had a few different places, but seemed to be very nuanced. And so I think both crews that came through were very focused on how we can better account for intestinal absorption, and all the technological strengths and gaps were highlighted. I think if you go into the details of the notes, the main sort of emphasis areas seem to be the lack of metabolism and the need to have what is called by some bioaccessibility. Susan talked about that in your session a little bit, about the ability of chemicals to be able to get into cells to then be available for first pass metabolism. And I think if you had to distill the entire session into one piece, it would be, having more rigor, more validated confidence in our ability to do first pass metabolism at different subsets, or using microphysiological systems as a sort of integrated system to do that. That would be my distillation.
Thank you so much. Lynn, did you want to add anything to that sort of high level?
No, I
summary?
thought that was a good summary.
Okay, all right, perfect, fantastic. Okay, well, I think in the interest of everybody's time, maybe we'll turn it over to Thomas now to close this out. And Susie, I don't know if you want to say any additional remarks. Okay.
Okay, good. So in the first hour of my wrap up, I will go through the key points and key no, kidding. You see how I think we can leave this meeting very happy even early because you saw it is impossible to summarize all of the things which were said in 2 times 10 minutes, which is a good sign because this was I think we saw a lot of things for discussion. It is certainly not a result of hallelujah, we have solved all of the problems. It is mapping challenges ahead, but I think it was also a very impressive show of opportunities. Especially the talks we had here showed the variety of models which have their advantages and disadvantages, which can be used and employed for some of the real life problems we have. We are certainly not always there to hand over from the shelf to a regulator tool to solve these things, but we also have to keep in mind these are young technologies. These are technologies which are, however, dramatically accelerating in their technical developments and what they have to offer. The main point I would like to make now is the way forward. The main goal of this workshop is to produce a report because we want to share with the outside world our assessments. This report will not be a consensus report. It cannot be because we are all individuals coming from very different views, but it will somehow characterize the mountain of work, the body of work which has been done in this field. Aspects of their assessment, we will make the respective disclaimers. We should normally everybody allowed to sign up to this report. Because we would like to present this as our joint assessment. Necessarily, you will not agree with each and every statement in this. There's always the possibility, obviously, to opt out of co-authorship at any stage. We will then offer acknowledging participation in the acknowledgement of the paper, but I also would not break out in tears if you decide not to want to be associated with the report, which would be a surprise to me after so much positive interactions here, but I'm just saying we are open for any of these. We're also aware that many of you will have to get blessing for these reports by the respective institutions. However, we have more than 40 times succeeded in getting these reports through, and we have almost always had regulatory agencies involved. So I think we know how to put the respective disclaimers that everybody is happy with this. The work here, the workshop was in part supported by the Transatlantic Think Tank for Toxicology, T4. It is a program I'm running out of CAT with my colleagues in the Center for Animal Testing. It is sponsored by the Dörnkamp-Binden Foundation, which is a philanthropic foundation out of Switzerland, which is about 40-something years in existence and is supporting the case of alternative methods. There is not really any strings attached to this. They're not taking any ownership over this, but they're promoting these type of exchanges. One of the deals we have with the Dörnkamp-Binden Foundation is that we publish the reports as a more or less default in alt-text. If there's any reason to deviate from this, we can do so, but I would not see any reason. The beauty of alt-text is that it is a full open access journal. The open access fee is going to be paid by the Dörnkamp-Binden Foundation. So the spread of this on your website, in your newsletters, and whatever is encouraged. We would like to see this. It contributes to the very favorable impact factor, which is around 6 over the last few years, several years. So it is one of the highest impact factor journals actually in the area of experimental life sciences and especially also biomedicine and toxicology. So we are really very happy that this journal is such a base for spreading our information. The process will be that Susan, who has been listening in and recording in all of these events, will develop the first draft, and after short check with some of the core team here, we will simply share this with all of the co-authors. You will then have the possibility to comment on whatever. We have a second round. In this context, we will be offering the co-authorship or opting out of this. If we feel that there is a need for specific contributions, things which we're a bit light on, we might ask some of you individually, could you write a paragraph here or add something? In some instances, we have even added co-authors when we felt out of the discussion or out of this product, there's something where we are not feeling comfortable to discuss, but there is an expert we could identify who might help us in saying something about the immune system and the gastrointestinal tract, for example. Yeah. So this is possible. It's unusual, but it is possible. The workshop report is not meant just as a documentation of what was discussed here. We are aiming for the best possible product. So if you have very inspiring ideas which you were not able to express during the workshop, we have no problem of something being added. It will make another round with everybody, so everybody has an opportunity to object, comment, or whatever. Our goal is to get this done latest within the year to publication. As soon as it is accepted after a peer review, it will be electronically available as a preprint. So it's a relatively fast process. Susan and I have discussed a goal of 2 months or so, plus minus, to get the first draft into our hands, and things are still fresh. So expect something as a Christmas present. So if you're bored under the Christmas tree, you know what you can do. Otherwise, any questions for me with respect to process? Normally, at the end of our workshops, we would have a round of feedback and also a round for discussing what other outcomes there are. We will not have this today, obviously, but I encourage that we, to some extent, think as the organizing committee of what are possible outcomes. This could include presenting this as symposia in some of the conferences like SOT, Eurotox, possibly others, the MPS World Summit. We could submit this as a proposal for a session, for example, which I think would be a very interesting one, as it is, from my perspective, the first attempt to see the actual regulatory utility and its challenges of a area of the use of microphysiological systems. I think this is really important. I think we can steer this out of the this coordinating group. There's no upcoming deadline. It's not tomorrow. Yeah, Eurotox is, I think, up in March and SOT in May. Then we also often discuss then possible activities, working groups, follow-up activities towards grant applications. In the interest of time, we will have to do this in centrally, but be prepared that we ask for possible engagement. Who's interested to join this or that as a follow-up of these activities. We will definitely include all of this, the online participants, as if they have been here on the table. So if this is so, no worries. Everybody we are inclusive in our approach. Again, does not matter who was here, which day and whatever, which time. If you are interested in participating, you're part of this game. That's it from my side. I think we can go home early and happily. It was a good exchange. I also enjoyed very much the interactions besides our discussions. I think this was a good group of people, and that's usually the recipe for success. So I'm looking forward to getting another report into the field. Thanks a lot from my side, and safe travels.